primary endpoint can be overall response rate. However, if you are using the regimen that's include combination of more than one medication, you need to have a randomization. And especially if with the era of the target therapy, when you are not expecting to have downsizing of the tumor, and all what you can expect is having disease stabilization. So single arm clinical trial here is not a good choice, and choosing a response rate is not a good choice, and you need to have randomized phase 2, and your primary endpoint should not be response rate. Mostly it will be progression-free survival. However, again, here a choice of progression-free survival between phase 2 and phase 3 is different. In phase 3 clinical trials, we need to compare median BFS or median overall survival or median disease-free survival between the experimental versus control arm, and this needs a large sample size. But in phase 2, it is time-driven or event-driven BFS. That's to say, 6 months rate of 6 months progression-free survival, 8 months progression-free survival. It depends on the situation. And here, when you are choice this 6 or 8 or 4 months progression-free survival as an endpoint, the sample size when comparing rate versus rate is smaller, and this is one of the criteria of having phase 2 clinical trial. So here, knowing the mechanism of action of the target therapy, whether it's going to have some downsizing or response rate or not, will affect the design of the trial, whether single arm or multiple arm, and will affect as well the choice of the primary endpoint. In addition, sometimes you have a tumor or that have a lot of molecularly defined subgroups. And the traditional way is to test for each subgroup and compare the patients having the biomarker versus patient does not having the biomarker. But if you have a disease like lung cancer, sometimes colorectal cancer that have a lot of subgroups, a lot of biomarkers, rather than having separate clinical trials with a long time to have data from each clinical trial, we have recent models of clinical trial which is quite important in the era of precision medicine. We have the umbrella or the basket design. What is the differences between these two designs? In the umbrella design, we are testing only single type of cancer. However, we have baseline testing of different biomarkers, and patients are allocated into different cohorts based on the positive biomarker. This is a classic flowchart of umbrella design of clinical trial. You have baseline testing of a set of biomarkers, and then patients with the first biomarker positive are allocated to cohort 1, another biomarker positive cohort 2, and etc. And in each cohort, you are comparing the new target therapy or new medication versus the standard of care. So this is the way of doing the umbrella design of trial, and it's a popular one in the era of having multiple biomarkers in the era of the precision medicine. So what is the advantage of having this kind of design? Actually, it is quite important to have expediting the process of the development of medication rather than designing separate clinical trials which need a long time. You can test all biomarkers with different cohorts, with different medication, all under the umbrella of a single trial. So this is quite important to expedite the development of new medication or new target therapies. And this is a growing list of umbrella trials in different tumor types like lung cancer, colorectal cancer, breast cancer, and others. And I will give one simple, I will highlight one simple example of umbrella trial in colorectal cancer. In this trial, patients who receive induction chemotherapy with FOLFOX plus Beva without disease progression after the course of induction therapy, and those patients were allocated into different treatment arms based on the biomarker available. For example, in BRAF mutant, the patients were randomized into maintenance, which is the control arm maintenance fluoropyrimidine plus bevacizumab versus fluoropyrimidine plus rituximab and vemurafenib. In the HER2 positive arm, patients were randomized into the control versus capecitabine plus trastuzumab plus pertuzumab, and this is a simple example of a kind of umbrella trial. Moving into the other type, which is a basket trial. Basket is a somewhat different. You have single molecular abnormality or single mutation, and you are testing the activity of the target therapy against this mutation in different tumor types, across different tumor types. And this is the this cartoon will show how to proceed. You are looking for certain abnormality, certain molecular abnormality, for example, in lung, colorectal, breast cancer, other tumor types, and you are testing a medication against this abnormality across these different tumor types. We have a lot of basket trials in the recent years. This is one of them, the Keynote 158. We are testing the activity of pembrolizumab in patients having high tumor mutation burden across different tumors, as you can see in this diagram. We have as well the NTRK inhibitors that have been tested against patients having NTRK fusions in different solid tumors, and the approval of medication like larotrectinib and others were based on this kind of basket trial with testing of this medication against patients having NTRK fusion across different solid tumors. Okay, this is some brief insights about the trials in the era of precision medicine, phase 1 and phase 2. What about conducting of phase 3 trials in the era of precision medicine? This is a classic design of phase 3. In the phase 3, you need to have definitive comparison between the standard of care arm versus a control arm. This is what we know. And traditionally, phase 3 trial should be randomized, should have a sufficient sample size to have definitive conclusion. How to conduct that in the era of precision medicine? The problem is you need to have a companion diagnostic, and you need to test the new medication against the standard of care based on the biomarker selected. And we have different ways of conducting phase 3 clinical trials in this era. You have the trial that is testing all comers and then looking for subgroups having the biomarker versus those not having the biomarker. Sometimes you can select for the biomarker from the upfront and exclude patients without the biomarker, and so we'll go briefly across different designs of clinical trials, phase 3 clinical trials in the era of precision medicine using using companion diagnostic or companion biomarkers. Let's look for the first one, which is testing for all comers. What does it mean? You are including recruiting patients. You are testing for the biomarker upfront, and patients were randomized. And then after randomization, you are comparing the experimental versus standard arm as a whole, and then you look for the comparison in the patients having the biomarker versus patients who are not having the biomarker. I will give you an example for that. Before that, there are two ways of doing that, for testing for all comers and looking for the biomarker. First, you can test the comparison. You can compare experimental versus control arm in the overall population, and then look for the subgroup having the biomarker. The other way is to have the comparison in the biomarker positive group, and then if positive, to look for the comparison in patients irrespective of the biomarker. I will give you a simple example, all of us I think know. In the Ambition 130 clinical trial in metastatic or advanced triple-negative breast cancer, those patients were randomized between the immunotherapy atezolizumab versus combined with nab-paclitaxel versus nab-paclitaxel. And please look to the endpoint. The endpoint is PFS and overall survival. First, they look to the intention to treat population, overall population of patients. And then if positive, they look for the comparison in patients having the biomarker, which is PD-L1 positive subgroup. So this is the outcome of the trial. In terms of statistics, in the intention to treat population, it is positive. However, it's not that clinically meaningful. However, if we in the PD-L1 defined subgroup of patients, we have clinically and statistically significant benefit of adding atezo to nab-paclitaxel. So first they looked to the overall population, and then they looked for the molecularly defined subgroup. However, we need to take that with extreme caution, especially in diseases who have multiple subgroups, multiple biomarkers, and so on. Why? Because if you don't have enough power for the molecularly defined subgroup from the start, you may end up by a trial that's not powered for the subgroup and you may have false positive results. So a better way to look for that is to have a design that like what we have been presented in the Keynote 355 study. Again, this is metastatic triple negative breast cancer. Patients were randomized between pembro plus chemotherapy versus chemotherapy. If you look to the hierarchy of statistical testing, they looked first for the comparison of the survival outcome in patients having the biomarker, which is PD-L1 CPS score more than 10. And if it is positive, CPS more than 1. And if it is positive, they look for the overall subgroup. So here we are making sure that we are having enough power for the biomarker-defined subgroup first, and if it is positive, we look for the intention to treat population. As we all know, we have positive results in the CPS more than 10% PD-L1 positive patients. However, in the overall cohort of patients, we don't have statistically significant finding. So it depends on the design. Either way is possible. However, in the intention to treat first design, you need to make sure that you have enough power for comparison in the subgroup of having the biomarker later. Another way of this retrial combined with biomarker is to test for the biomarker upfront, excluding patients without having the biomarker and only randomizing patients positive for biomarker. The classic example for that is a well-known HER2 positive breast cancer trials. They are testing for a phenotype of patient upfront. Patients with HER2 negative are excluded, and patients with HER2 positive are included. Another recent model for that is the responder trial in hormonal receptor positive HER2 negative breast cancer. They are patients after surgery having 1 to 3 lymph node positive were tested for Oncotype DX. Patients with recurrence score more than 25 were excluded. This is of the study. Patients with recurrence score from 0 to 25 were randomized between chemotherapy plus endocrine therapy versus endocrine therapy alone. So they are excluding patients without the biomarker of interest from the start, and this is a result. All of us know that. In post-menopausal, we don't have difference between adding chemotherapy versus to hormonal versus hormonal alone. However, the benefit is restricted of adding chemotherapy is restricted to premenopausal patients. Another example of phase 3 trials in the combined with testing for biomarker is, again, it is all-comer design. However, they are testing for the biomarker upfront, and they have separate randomization. For the patients with biomarker positive results, they are randomized separately into experimental versus control arm. Biomarker negative randomized separately in a separate randomization between control and experimental arm. And we have some famous example for that. This is one of them in the SOLAR-1 trial in breast cancer. Patients with hormonal receptor positive HER2 negative who have metastatic disease, received at least one line of hormonal therapy before. They are tested for BIG3CA mutation. In the mutant cohort, we have randomization between alpelisib plus fulvestrant versus fulvestrant, and we have similar randomization in the patients with mutation negative cohorts. So this is the result. We have benefit in the mutation positive cohort, but we don't have benefit with patients without the mutation. So we are doing sometimes that to have proof of the concept that the medication activity is restricted to the group having the mutation, but no benefit at all for patients without having the mutation. So it is quite assuring that you need to select patients having the mutation alone. So again and again, you have different design. You can choose whatever design that's feasible. It depends on many factors. How confident is the biomarker? How confident is the link between the biomarker and the activity of medication? For example, in HER2 positive, we know from a lot of data before that it's not working in patients having HER2 negative disease. So we don't have to test this medication in HER2 negative patients. That's why we are excluding them from the start. However, for a new biomarker that we don't have that confident, like alpelisib in the SOLAR-1, we need to have randomization as well in biomarker negative subgroup to prove the concept that the medication is working alone in patients without the biomarker, patients with the biomarker alone. Another and the last one that I will address in my talk is a somewhat more complex design in the area of precision medicine, which is adaptive clinical trials. In adaptive clinical trials, you have from the start, you don't know exactly how the medication is working against the standard. So you need to have certain assumptions from the start, and you need to have certain interim analysis, and you can modify the design of the trial while the trial is still ongoing. You can modify the sample size. You can modify the allocation between groups from randomization 1-to-1 to 2-to-1 or something like that. Sometimes even you can modify the endpoints. So you can modify the trial according to the results of the interim analysis. And you are doing that if you don't have enough clinical data before about the activity of your recent medication or your target agents against the standard treatment for the disease you are testing for. And we have expanding list of these adaptive clinical trials over recent years. And even sometimes you don't have two-arm adaptive trials. Sometimes you have multiple-arm, multiple-stage adaptive clinical trials. This is very complicated trials. You have certain interim analysis at well-defined points. This need to be well-defined from the start of the trial. And according to the results of the interim analysis, you are comparing the multiple treatment arms against control. Some treatment arms who are doing poorly are removed while from the first interim analysis, and then you are going for the second. You may remove another arm that's not performing well. And at the end of the study, you may end up by selecting one or two new medication to be tested against the standard or control arm in further trials. To make it more simple, please look to this diagram. In the left side, this is the traditional way. If you are comparing in a separate trials, experimental arm against control arm, experimental arm against control arm, in separate clinical trials. But if you want to expedite the process, in a single trial, you are comparing multiple experimental arms against the control arm in plaque. In the first interim analysis, the medication number 2 is not working well. Is not performing well according to the assumptions of the trial. So it is removed. And then you are going for the second stage of the trial. Medication number 3 is not working well, so it is removed. And then you end up by two experimental medication that have promising results against the control arm, and these medications are selected to be compared in phase 3 clinical trials. So it's a kind of complex design, but it will expedite the process of development of medications. So this is my last slide. So again and again, basically, the steps of comparing or testing the new medication in the era of precision medicine, basically, it is the same steps, phase 1, 2, 3, 4. However, we have some differences. In phase 1, it's not essentially the dose selected to be maximum tolerated dose, and you need to develop companion diagnostic from the start. In phase 2, you have basket and umbrella trial design, and sometimes you have adaptive clinical trials. And in phase 3, you need to have enrichment by biomarkers, either testing for all comers or excluding patients without the biomarker from the start, and you will choose a proper design according to the situation. And thank you very much for your kind attention. Thank you very much.
Thank you so much, Dr. Sharif. Okay, so moving on with our next speaker, going to be Dr. Riham Ajina. She's an assistant professor at King. You can't hear it? Okay, so thank you so much, Dr. Sharif. Moving on with our next speaker, Dr. Riham Ajina. She's an assistant professor at King Saud University for health science. She's going to talk to us about immunotherapy biomarkers. So let's hear from you, Dr. Ajina.
So thank you for the introduction, Dr. Faiza, and thank you for the organizers for the invitation, especially Dr. Kanaan Shamari. Before talking about or indulging in the presentation about immunotherapy biomarkers, I would like to remind everyone of how cancer cells can recognize, or how the immune cells can recognize and kill cancer cells. Can you hear me? Okay. So the first step, cancer cells typically have a lot of mutations. Some of these mutations will be translated into peptides. Because these peptides are different than normal cells, they will be recognized by the immune system as non-self, so they will be considered as tumor antigens. Then antigen-presenting cells will engulf these tumor antigens and process them and present them on the surface of antigen-presenting cells. These antigen-presenting cells will then migrate from the cancer tissue to the secondary lymphoid organs. In the secondary lymphoid organs, then they will interact with naive T cells. So T cells that have never been exposed to antigens before, but have specific receptors that can specifically bind to the tumor antigen expressed on the surface of antigen-presenting cells. Those naive T cells then will get activated, and then they will migrate through the blood vessels toward the cancer tissue. In the cancer tissue, if those T cells have receptors against that specific tumor antigen expressed on the surface of cancer cells, they will be able to recognize and then kill this cancer cell. When the cancer cell will die, you will get more tumor antigens, and thus the propagation of the cycle. Because these tumor antigens are different than the normal cells, they are sometimes called them the neoantigens. This is how our immune system can recognize and kill the cancer. But the cancer cells are very smart, so they tend to try and avoid the immune recognition and destruction. For example, with the immune system or the cancer cells can reduce the expression of the cancer or tumor antigens on the surface of antigen-presenting cells or cancer cells. If there's no tumor antigen, then the T cells will be blind to the cancer, and then they will not be able to recognize and to kill the cancer. The other immune evasion mechanisms are the immune checkpoint inhibitors that everyone knows, which are, for example, the CTLA-4. Sorry. So the CTLA-4, I don't think you can see it. The CTLA-4 here, which will inhibit naive T cell priming and activation, and the PD-1 and PD-L1 signaling that will inhibit T cell anti-tumor activity. Because we know these immune evasion mechanisms, the immunotherapy was developed to overcome these immune evasion mechanisms. For example, the anti-PD-1, PD-L1, and anti-CTLA-4s. There's also the CAR T cells, which can basically just provide the body with more T cells that are capable of targeting and killing the cancer. There's also the cancer vaccines, which is basically providing the body with more antigens and immunogens to stimulate tumor antigenicity and immunogenicity. The currently approved immunotherapies, or immune checkpoint inhibitors, are the first one was the ipilimumab for CTLA-4, then we have 4 for PD-1 and 3 for PD-L1. The most recent one is the anti-lag that was approved last year in 2021. So we know that cancer immunotherapy, cancer immune checkpoint inhibitors are very effective. However, we also know that most patients do not respond to immune checkpoint inhibitors. For example, if you see in this clinical trial that was published in New England Journal of Medicine 2019, patients with advanced melanoma disease, when treated with immune checkpoint inhibitors, the 5-year survival was for ipilimumab 26%, for nivolumab was 44%, and for the combination of nivolumab plus ipilimumab was 52%. This is a good number for patients who have advanced disease, so they were expected to die. However, we need to increase this number. We need to improve the number to cover all cancer patients. And we need to have predictive biomarkers to know who's gonna respond, who's not gonna respond to the treatment. So the currently approved immunotherapy biomarkers are 3. The first one is PD-L1 expression. The second one is mismatch repair deficiency or microsatellite instability. And the third one is tumor mutation burden. The first one is PD-L1 expression. It makes sense. If you have more of your target and you're targeting PD-1 or PD-L1 signaling, then if you have more of your target, you would expect the treatment to work. However, although there were more than 46 trials that were conducted, Most of these studies, for example, 5 in melanoma, 4 in renal cell carcinoma, 7 non-small cell lung cancer, and so on. Most of these studies showed that there's first inconsistency. And the second thing is that there, in fact, PD-L1 expression is not very significantly, it's not a very good biomarker. In other words, if you have PD-L1 expression, it doesn't mean that the patient respond. And even if there's no PD-L1 expression, the patient still may respond to the treatment. The second biomarker is mismatch repair deficiency and microsatellite instability. So what are mismatch repair proteins? They are a set of proteins that function by correcting the DNA. So if there is a DNA damage, these proteins work to correct this damage. If there's deficiency, so these proteins are not functioning or they are not there, then patients with mismatch repair deficient proteins will have more DNA damage, will have more mutations. In fact, they found that DMMR tumors have 10 to 100 more times of mutations compared to mismatch repair tumors with mismatch repair proteins abnormal level or PMMR proficient. Also, these mutations typically occur in the microsatellite regions. Therefore, it's typically known that DMMR tumors, also known as microsatellite instability or MSI high tumors. And as you recall from the cancer immunity cycle, we said that if there's more mutations, some of these mutations will become immunogenic, and thus the immune system will be sensitized. With that hypothesis, several clinical studies, for example, these two that were reported in 2015 and 2017 by Dr. Lee, it has been shown that, for example, in this clinical trial, 86 patients who are all DMMR, they were recruited to the treatment. They all have, to the clinical trial, they all have advanced disease and across 12 different types of cancer. In this study, it was interesting to see that when these DMMR patients treated with anti-PD-1, 53% of the patients had objective responses, and 21% of the patients had complete responses. Again, these are advanced disease. As a result, this biomarker was FDA approved. And the third biomarker is tumor mutation burden. Going back to the same concept, if you have more mutations, you would likely to see more tumor antigens, thus the patients will be more responsive to immunotherapy. So scientists and physicians decided that maybe we just directly look at the tumor mutation burden. And in many clinical trials, for example, the three ones are shown here, in colorectal cancer, urothelial cancer, pancreatic cancer, patients who have high tumor mutation burden, shown here, so high number of frequency of mutations in green and in blue, had a better overall survival compared to patients with low tumor mutation burden. This again led to the FDA approval of this tumor biomarker. The next step would be, what is the relationship between the TMP biomarker and the MSI biomarker? And several studies have been conducted to answer the same question. For example, the two shown here, the one on the left shows that 62,000 samples were included in the study. Among these, only about 4,000 of them were at high tumor mutation burden. And only a smaller subset shown here in gray had MSI high. What I want you to consider here is that there is a great overlap, but it's not 100%. So there are some patients who are MSI high, but they were not TMP high. The same exact result was reported the year after, so in 2018, by a different group with even a larger number of samples. So how to move that into the clinic? In 2019, there was a very nice clinical trial that was presented in ESMO. In this clinical trial, they took 22 patients, MSI high, metastatic CRC colorectal cancer patients. They stratified those patients into high tumor mutation burden and low tumor mutation burden. Interestingly, they found that patients who are MSI high and TMP high, shown here in pink, all were complete responders and partial responders. Whereas patients who had low TMP, still they are MSI high, but they have low tumor mutation burden, they all had stable disease or progressive disease. That was also represented by the overall survival rate. where the orange here shows the tumor mutation burden high patients. So that was very exciting result. What I want to show you on this slide is the most recent data we have from the last week from ESMO 2022. In this study, so for all the previous studies, we had patients who have advanced metastatic disease. In this study, they included patients who have non-metastatic disease. So non-metastatic DMR colorectal cancer patients. And there were 112 patients. These patients were treated with nivolumab and ipilimumab 6 weeks prior to the surgery. So these were neoadjuvant therapy. Interestingly, 99% of the patients had a pathological response, with a major pathological response of 95% of patients. 67% of patients had complete responses. The toxicity profile was very acceptable, with only 4% of patients had grade 2 and grade 3. But we have to stop here and think about the challenges. The first thing is that those patients who are both MSI high and TMP high are very rare, as we saw in the previous figure. The second thing is that there are so many technical challenges. As a scientist, I face these challenges every day. Some of them, these technical challenges, is the quality of the sample you get. So is the DNA pure? Is it degraded? Is it not degraded? Then which sequencing panel should you pick? Should you pick a very comprehensive panel, spend much more time, money on it, or get a smaller panel that's less comprehensive? Which one is more representative to the whole genome? This is a question that is very difficult to answer. And then there's the bioinformatic analysis. There's so many tools. There's the difficulty of deciding what is the cutoff, when to say there is a high mutation burden, moderate, or low mutation burden. This is something that, is it going to be the same for all types of cancer, or is it going to be a cancer type specific? To answer this question, there was a project, the TMP harmonization project, started in 2014. They are trying to basically answer these technical problems. In this project, there were so many people involved. For example, the US FDA, the National Cancer Institute, so many different companies such as AstraZeneca, Genentech, Foundation Medicine, Illumina, Pfizer, Thermo Fisher, QIAGEN, and Roche. So it's a huge project. And the take-home message is that they are still trying from the last 8 years to just solve these technical problems to improve the quality of the TMP biomarker. The last challenge is the tumor heterogeneity and complexity. And I would like to spend a few minutes here, because as a scientist, again, I see this in my preclinical studies. If you take a group of patients, you know that every particular patient will have a unique tumor. If you look at one patient, every metastatic tumor will be different. If you just take a biopsy of the sample, every single cell will be different than the cell adjacent to it. And that differences, these differences will be at the genomic level, epigenomic level, the proteomic, phosphoproteomic, metabolomic, all these changes, all these components of the cell will be completely different between one cell and another. Also, within the same cell, if the cell has two alleles, each one of these alleles might have a different mutation of the same gene. Despite all this complexity and heterogeneity of the cancer cells, we don't only have cancer cells, but we also have cells surrounding these cancer cells, which is called the tumor microenvironment, and it's very complex. In fact, if you just look at the tumor under the microscope, you would find that cancer cells account for less than, sometimes in some cases, about just 1% of the whole tumor, and 99% of the tumor is just tumor microenvironment. So what are the components of the tumor microenvironment? We have many immune cells, such as tumor-associated macrophages, MDSCs, dendritic cells, mast cells, tumor-associated neutrophils, NK cells, B cells, and T cells. Each one of these cells have different types and even subtypes, and they can change from one subtype into another. We also have neuronal cells, fibroblasts, adipocytes, lymphatics, blood vessels, altered microbiota, and altered microbiome. We also have the non-cellular components of the tumor microenvironment. And what is very important to know is that the maturation stage and activity profile is changing every second. And also each one of these cells, so we have hundreds of or thousands of different components, each one of them is directly and indirectly interact with each other. Knowing all that, the question now is how can we study a tumor that is complex, heterogeneous, and dynamic? I think the answer is going back to the same pictures. We need to study every patient. So doing experiments N of 1, think about or trying as much as possible to study all the cells, but we know it's impossible. So we need to get a representative sample of the tumors. We also need to consider all components of the tumor microenvironment. We need to do that at the genomic, epigenomic, metabolomic, and proteomic levels. We also need to consider the disease burden, the performance status, the history of autoimmunity of the patient. And because cancer is very dynamic, every time the patient will have progressive disease resistance, we'll need to repeat all these multi-omics, very complex experiments again. Does that really happen in reality? I will give you just one example. So in this PRINCE phase 2 clinical trial for pancreatic cancer patients, they stratified 105 patients into 3 groups: control plus nivolumab, chemo plus nivolumab, anti-CD40 plus chemo, and anti-CD40 plus chemo plus nivolumab. Despite the very promising result in phase 1, phase 2 was negative. But this paper was published in Nature Medicine because they did very comprehensive translational profiling. For example, so they took from 105 patients, they took tumor samples, they took blood samples, and stool samples. Just from the tumor tissue, they did Victra panel, which is basically multiplex immunofluorescent Victra panel. And this is one of the very promising and very emerging tools that it's expected to be approved for the use of as an immunotherapy biomarker. So in this assay, you would select 9 biomarkers that you can stain with immunofluorescence simultaneously. Your tissue will look like this. And then if you zoom in, you will start to see more colors. If you even zoom in, you would see each single cell. What are the markers that can be expressed by this cell? With this, you would know what is the geographical location of each one of these cells. For example, do the T cells that are active or express PD-L1 are adjacent to cells, the cancer cells, or they are just at the marginal surface of the tumor? Going back to this figure, so from the tissue, they did this Victra panel. They also did sequencing. So they did whole exome, they did RNA-seq, and T-cell receptor sequencing. When I say T-cell receptor, it's just for the T-cells within the tumor. They did the sequencing for their T-cell receptors to know the clonality and to know what are the antigens that were targeted by the tumor. They also did, for the blood, they did cell-free DNA. They did to study the mutation of KRAS. They also did proteomics. They did PPMC profiling to study the immune profile. So the immune profile within the blood, the immune profile within the tumor. And they did the microbiome analysis. As you can imagine, this is a huge amount of data. As a physician, how can you find the needle? How can you find the clinically applicable data? I think the answer is you need scientists. So you need scientists, for example, bioinformatician, to give you their magnets, their tools to help you pick up the needles. And that's what they did in this exact clinical trial. So they took all that raw data, processed the data, stored them in Candle database. And then with the help of bioinformaticians, they were able to integrate all these very complex multi-omics data to identify tumor biomarkers that can help them identify new therapies, better patient selection, and for new clinical trials. I just want to end here by saying one sentence. This was taken from the Candle database website. The Candle analysis platform designed by data scientists, immunologists, engineers, translational medicine experts. So we need to work hand in hand from both the medicine and the scientific fields in order to improve our patients' outcome. I know that having a balance between comprehensive multi-omics data could be not clinically applicable, but having that balance is very challenging. Therefore, we need to have biomarkers that are specific, sensitive, not invasive, cost-effective, give us rapid turnaround time so the patients will not die before we do all these omics and integrative analysis, and give us very easy to decide or assist in making the right medical decision biomarkers. Is that possible? I would say what exactly Dr. Robert Schreiber, an immunologist and pioneer in the field of cancer immunology, said. We are at the beginning of the end of cancer, but certainly not the end of cancer. So we have a lot of work ahead of us, but at least now we are working with great expectations of things to come. Thank you.
Thank you so much, Dr. Gina. This was a very interesting and exciting talk. So moving to our next speaker, Dr. Marwa Derwish. Dr. Derwish is going to talk to us about microbiome in cancer and its therapeutic potential. Welcome, Dr. Derwish. Assalamu alaikum. Thank you, Doctor, for introducing me. I would like to thank the organizing committee for inviting me to this prestigious conference. It's my pleasure to be one of the speakers in this conference. So as you can see, the title of my talk is about microbiome in cancer, therapeutic potential. So since there is a growing evidence in the literature discussing the role of the gut microbiome in colorectal cancer development, so I will be specifying my talk about the colorectal cancer. Would like to start my presentation about some epidemiological data about the incidence of colorectal cancer. So as you can see from the worldwide map here, colorectal cancer accounts for about 10% of all annually diagnosed cancers and cancer-related deaths, and the most and the highest number of colorectal cancer is in developed countries, and the stabilizing rate in colorectal cancer in developed countries is due to the application of a nationwide screening program for colorectal cancer. However, the trend is increasing in developing countries, and the incidence of colorectal cancer is predicted to increase to 2.5 million new cases in 2035. Now, for Saudi Arabia, according to the International Agency for Research in Cancer, colorectal cancer considered the first among males and the third one among females, and that's due to the lack of presence of screening programs in Saudi Arabia. So there is a need for screening program. However, the available screening tests lack sensitivity, specificity, or are associated with considerable burden, risks, and costs. So data from the literature suggests that the gut microbiome might be related
to the risk of colorectal cancer. And here are some studies conducted in order to study and to investigate the role of the gut microbiome in the development of colorectal cancer. Some authors, some people interested in identifying the potential of fecal microbiota for early stage detection of colorectal cancer. On the other hand, other groups interested in studying the gut microbiome as a promising biomarker for immunotherapy in colorectal cancer. So we hear a lot about microbiota and microbiome. What does it mean, microbiome? What does it mean, microbiota? The human microbiota refers to the microorganisms that are available or found in a specific environment in the human body. So we refer to the microorganisms that are found in the large intestine to be the gut microbiota. However, the human microbiome refers to the collective genomes of all the microbiota present in that habitat. And these microbiota includes archaea, bacteria, viruses, parasites, and fungi. However, the most studied member of gut microbiota is bacteria. So looking at this slide, you can see the distribution and the diversity of gut microbiota along the gastrointestinal tract. So starting from the stomach and ending to the large intestine, we can notice that the quantity of the gut microbiota, as well as the diversity of microbiota members are increasing. So given that the colon consists of the largest number and the highest diversity environment of microbiota in human body, and that's why there is a strong evidence in the literature associated the microbiota present in the colon to the development of colorectal cancer. And the figure on the right-hand side describes the different roles of gut microbiota. Gut microbes in our human body are present in either two situations, either in a homeostasis situation in which there is a balance between a normal gut microbiota, and here the gut microbiota express beneficial effect to the human body, and mostly in the regulatory functions, where they keep where microbiota keep a normal healthy epithelial barriers in the large intestine. However, when there is imbalance in the gut microbiota in the human body, the situation we call it dysbiosis. So there is a pathogenic effect of the gut microbiota in this situation, and might be contributed to the development of different diseases such as inflammatory bowel disease, Crohn's disease, and colorectal cancer. So here in this slide, we can see the modifiable and non-modifiable factors that affect the gut microbiome in our bodies. So we'd like to specify here the modifiable factors, so something we can modify it, okay? So we have the medications, the diet, especially the fiber-rich plant-based diet, which have a positive impact on the gut microbiome versus typical Western diet that have a negative impact on the gut microbiome. Also, the environment, including geography, cultural preferences, environmental exposure, psychological factors, all these modifiable factors can influence the gut microbiome either positively or negatively. However, what are the colorectal cancer risk factors? We can find some of similarities between the risk factors that are responsible for colorectal cancer and the modifiable factors that influence the gut microbiome. Example of modifiable risk factors that increase the susceptibility of individual to colon and rectal cancer development: processed meat, red meat, low intake of vegetables and fruits, body fat, and obesity, and all these factors as well influence the gut microbiome negatively. However, the modifiable risk factors that decrease the risk of colorectal cancer include physical activity, dietary fibers, dairy products, vitamins, calcium, and these as well influence the gut microbiome positively. So what are the methods of microbiome modulation? How we can modulate the microbiome? According to a review published in 2019, there are either of 4 ways that we can modulate the microbiome, either through the fecal microbiota transplant, and each one of these methods have its own advantages and disadvantages, and considerations should be taken in order to implement that method. So starting with the fecal microbiota transplant, we have to consider the donor selection, the delivery mechanism, the need for conditioning regimen, and then the second method is probiotics and bacteria consortia, in which it's easy, affordable, and also accessible. However, there are also very important considerations in terms of use of spores versus live bacteria. The third method that we can use in order to modulate the microbiome is prebiotics, which refers to the fiber supplements itself of the beneficial bacteria. And then last but not least, the diet. So holistic, which the advantages of using a diet to modulate the microbiome is a holistic change that might have other health benefits. So here are some open questions regarding the role of the colonic microbiome in the development of adenomas and in the different stages of colorectal cancer, starting from the dysbiosis, adenoma, to preclinical cancer, clinical cancer, and then death. Here are 4 different questions that we have to answer in order to make a significant contribution to this early screening and detection of colorectal cancer. So for each individual, a gut microbiome index might, a risk personal index might be generated based on the gut microbiota data, based on the genetic factors of that individual, a diet and a lifestyle. So how we can answer these questions? We can answer these questions by using the multi-omics approach or the multi-omics analysis, which includes analyzing high-dimensional biological data from the microbiome sample, its environment, or from the host, which includes starting with metagenomics, studying the gene content and mutations, and then moving to the metatranscriptomics, in which we have to study gene regulation and expression, and then the metaproteomics, in which we study protein content, activity, and post-translational modification. And finally, we study metabolomics, which refers to the endogenous and exogenous metabolite presence and fluctuations. So in order to have a holistic understanding of the role of the gut microbiome in colorectal cancer development, we have to study the tumor microenvironment from different perspectives. So to starting with a metagenomics approach in microbiome studies, this schematic diagram represents a workflow for analysis of a targeted and shotgun metagenomic sequence data. In a targeted metagenomic sequencing, we are targeting the conserved gene 16S ribosomal rRNA gene, which is a conserved gene in all bacterial species. However, in shotgun metagenomic sequencing, we do the whole genome sequencing of all the microbial community available in the microbiome sample, studying their metabolites and their function as well. So data from the literature identified the microbiota associated with colorectal cancer in humans, some of them gram-negative, some of them gram-positive anaerobic bacteria, and found different in relative abundance of those bacteria in colorectal cancer patients than in normal individuals. So what's the current situation in Saudi Arabia in colorectal cancer in regard to the microbiome? Unfortunately, there is a lack of microbiome studies in Saudi Arabia, despite a high importance and increasing trend in microbiome studies and its role in colorectal cancer. And there is also a lack of colorectal cancer screening programs in Saudi Arabia. So we as scientists, we as clinicians, we have to contribute to the implementation of Saudi 2030 vision in terms of vibrant society and improve the quality of patient lives. So initiative has been taken with collaboration from King Abdullah International Medical Research Center, King Abdulaziz Medical City, King Saud bin Abdulaziz University for Health Sciences, King Abdulaziz Medical City for Science and Technology, and we do have an international collaboration with the University of Leicester in the United Kingdom in order to start implementing microbiome study in Saudi Arabia and investigate its role in colorectal cancer development. So the study area in this slide would like to explain the study area. It's in the Ministry of National Guard Health Affairs central region between the university, collaboration between the university, the research center, KMARC, and King Abdulaziz Medical City. So as for the study participants, we do have 3 groups of participants. The first group is about naive colorectal cancer patients. And we refer here to naive because we are recruited colorectal cancer patients who are recently and newly diagnosed with colorectal cancer before any intervention, before chemotherapy, before the surgery, before the radiotherapy, because we want to study the microbiome as it is without any intervention. And we do have 2 control groups, the patient's relatives. So we ask the relatives who are with the patients before the surgery in order to provide a stool sample. And we do have also independent healthy volunteers as control group for different purposes. One of them in order to do the validation of the data, and as well as in order to have another control group, since we don't have any microbiome data in the Saudi Arabian population. The study is ethically approved by the IRB office in KMARC, and we do have the consent forms, so the patient and the relatives consent to contribute to the study, and we collect the food survey data from the patients and the control group, since there is an evidence of a diet impact on the gut microbiome. So far, sorry. How can I go back? Yes, I think, yes, sorry, it's fine. So we are collecting, because since we are, this is a first step, so we are like building a platform for microbiome study. We are collecting at first stage stool samples from each participant. So far, we do have 52 colorectal cancer patients. So we do have a stool sample from 52 patients. From patients relative, we have 36, and from the independent group, we have 45 stool samples, and we are continuously collecting samples from patients and control groups. We do have some exclusion criteria that have been shown in the literature to influence the gut microbiome. So we excluded patients who received antibiotics. Antibiotics influence hugely the gut microbiome. So we don't want any, we excluded patients who received antibiotics within last month of specimen collection. We do have also excluded patients who have a personal history of cancer, and patients with chronic viral hepatitis or HIV, or patients who do have their cancer due to genetic causes or due to a family history. We excluded those patients. We also excluded patients who are on radiation or chemotherapy for their current colorectal cancer. For the control groups, we do exclude patients who again received antibiotics, who had colorectal cancer before, and who had colonic abnormalities or gastrointestinal tract disorders. We for sure excluded pregnant and lactating women from participating in the study. So for the fecal specimens workflow, we instructed patients and controls how to collect stool sample, and the stool sample is collected in a special designed fecal collection tube, specifically designed for microbiome study, and then we do the microbiome DNA extraction. And then we prepare the library for the 16S rRNA sequencing to do the targeted metagenomic analysis. All of this work is performed at KMARC. So as for preliminary results, in order to do like a piloting of the study, and in order to optimize the experimental conditions, we didn't want to risk all the patients and consume all the samples to do the experiments. So we started by randomly selected a number of stool samples from each group in order to start optimizing the experimental conditions. So as you can see, from the cancer patient, we selected 7. From each of the control groups, we selected 8 individuals. And these are the initial demographic data in regard to the age, body mass index, gender, hypertension, and diabetes. So these are the initial metagenomic diversity analysis between the naive colorectal cancer patients and healthy volunteers. the data is still in its initial stage of analysis, and a lot of effort has been made in order to complete the analysis and to do further investigation, and in order to compare our results with the results available in the literature. As you can see, there are a variety of microorganisms, specifically bacteria, available in each of patient sample, and there is also differences between the naive cancer patient and healthy volunteers. There is also a difference between the naive colorectal cancer patients and healthy volunteers, which is the independent group. So the ongoing work right now at KMARC, at King Abdulaziz Medical City, and at the university is to complete recruiting patients and healthy volunteers to the study, and also to prepare for targeted metagenomic analysis for available fecal samples, and to do complete analysis, bioinformatics analysis, to do comparison with the data available in the literature, to investigate whether a Saudi Arabian population have a different microbiome profile, different than an international data. Here, I would like to thank all the study team and the supporting team at KMARC who contributed effectively in this study. Without their efforts, this study cannot be started and cannot be completed. So at KMARC, I would like to thank Dr. Majid Al-Raibi, Head of Infectious Disease Department, for his significant contribution. Ragad Al-Aqil, she is responsible for recruiting samples and do initial analysis, experimental work from King Abdulaziz Medical City. Thank you to Dr. Mohammed Al-Girni and Dr. Ayoub Al-Girni, Dr. Nahar Salim and Dr. Hamoud Raufi for their significant contributions and help providing us an access to patients and the relatives in order to collect samples. And then COX, we do have Dr. Musa'ad Tamimi, University of Leicester, Dr. Primrose Freeston, she's working as a consultant with us, and then the supporting team at KMARC. Thank you very much.
Okay, thank you for the excellent presentation. I would like to ask the speakers actually to join us over here first. Dr. Marwa, over here, Dr. Rehan and Dr. Kanan. So also I would like to ask our audience, actually online audience, to submit their questions in the question and answer box, and we'll go over them one by one. Okay, so Dr. Fahad Al-Maqbil with us. He has a question.
Thank you all. It's really great session, great session. Thank you all. So all the great four speakers, the excellent starts for this great conference. My question to Dr. Rehan regarding last year in ASCO, there was a paper about melanoma and immunotherapy. They received it in the morning compared to those patients who received late. And the response is more for better for those patients who received the immunotherapy in the morning. Do you have any explanation from science point, like why that circadian rhythm different may affect the response for the patient with immunotherapy?
Do you hear me? Is it working?
Yes.
Okay. So I'm not going to answer for that exact particular group. So, do you hear me now? Is it working? Okay. So I'm not going to answer that for that exact particular group because in fact, I actually don't know the answer. But what I know is that there was a paper published recently by a group in a good journal. And what they found is that even interpreted sleep, so if the patient woke up and slept, that can induce tumor growth in patients just because the tumor microenvironment, particularly tumor-associated macrophages, will be more active somehow, we don't know how, through some signaling pathways that the tumor microenvironment will induce tumor growth. So I guess it would be something like that. It's just the tumor microenvironment, some molecular mechanism. I guess that the question then would be, is it really very important to know this particular mechanism, and then can we target it? Because then if you think about it clinically, are we going to just treat the patient in the morning versus the night, and how are we gonna do that? So again, it's a great question because it highlights why the tumor microenvironment is complex and why we need to do all these multi-omics studies. Thank you.
I have a question to Dr. Sharif, as well as to all our speakers. Do you think AI might help actually in developing more information about the cancers and treatment?
Thank you.
Biome.
It's Sorry. Is it clear now? Okay. So thank you very much for your question. Is the AI? Yeah. So would you please repeat your question?
Is there any study like that?
The artificial intelligence has been recently applied in microbiome studies, in which the microbiome, the artificial intelligence holds all the microbiome data and help in predict the possibility of a certain disease. But in order to implement the AI in microbiome studies, we have to build a database for microbiome data. And as I mentioned in my talk, that the microbiome data is completely different between each country. So once if it's applied in, for example, Western societies, we have to implement that in Saudi Arabia because we do have our own microbiome. So we need first to build a microbiome database, and then we can implement these data in AI in order to give us a prediction about a specific disease. Yeah, thank you. Okay, so we have a question back also for Dr. Marwa. Okay. So which method, you mentioned in one of your slides that there is different method to do microbiome or to enhance microbiomes in our body. Is there a preferred method or better method, safer method than the other, or are they all pretty much do the same thing?
Okay, that's a great question. Thank you very much. Each method has its own advantages and disadvantages. But for any microbiome study, the initial method is the first method that we have to start with is about metagenomics. And that's the thing we have applied in our study. Metagenomics itself can be applied either targeted or non-targeted. The targeted one, if you want to target a specific gene, and this will give us only the bacterial community in a microbiome sample, that's the 16S ribosomal rRNA. This method has its own advantages, has its own disadvantages as well. However, the shotgun metagenomic analysis that we sequence all the microbial community available in a microbiome sample and the metabolites produced by this microbiome and study the functional analysis of this microbiome is a more comprehensive and more precise approach in order to study the microbiome. However, it's highly expensive, and it needs a lot of work, a lot of bioinformatics capacity in order to do the analysis. So the approach that we are using, that we are starting from the baseline. So we starting from 16S rRNA data, and then we can pick some of the samples in order to do the shotgun metagenomic analysis. This is the smart way how scientists around the world, they use the microbiome analysis. We cannot jump to the whole genome sequencing without knowing the 16S rRNA data. So it's like a sequential step, and we have to judge wisely which method to start with and which method to complete with.
Okay, great. Thank you so much. Dr. Mohammed Al-Garni has a question.
Okay, so thank you all for this great talks. My question is to Dr. Riham. So for patient with colorectal cancer, the same biomarker that we use in early stage, MSI, for example, the study that you present, the NICH2 study, we have a better result and better outcome compared to the metastatic setting, although we are using the same biomarker. So my question is this biomarker static, or is there any change that happened during the course of the patient that lead to a different outcome?
So I think that clinical trial will hold. So it was just started for, or was followed up for 1 year. We didn't know what would happen after 5 years. I could be wrong, but I expect that now 99% of the patient benefited. I would expect that some of the patients will have relapse. Those patients would have relapse, then we will need to further investigate and to know what is the mechanism of resistance. But for now, it seems effective at least for all except 1%. And I expect that there will be some dynamic and resistance that will happen in the next few months. Hopefully not for the patient, but that's what we have been seeing in the investigation.
Thank you very much. So I have a comment and question for both you, Dr. Marwa, and Dr. Riham. So the first one is for you, Dr. Marwa, which is basically related to the microbiome. So I published an abstract with the GI group in National Guard with Dr. Mohammed Al-Garni on hepatocellular carcinoma. So hepatocellular carcinoma is a disease that is unfortunately prevalent in our society. We treat it with immune checkpoint inhibitors. And what we've done is we retrospectively looked at ones that were given antibiotics versus ones who were not given antibiotics. And I have shown a statistically significant association that showed that the survival of ones who were given antibiotics concurrently with immunotherapy was significantly less compared to the ones that did not take antibiotics. And my question to you is, how can we utilize this piece of information to have some sort of therapeutic impact, other than, of course, not giving antibiotics to our patients? The second one is to Dr. Riham. I really liked your slide about the non-concordance between MSI high and high TMB cases. And there will be a debate between myself and Dr. Mirvet right at the end that talks specifically about this. And my point is promoting doing next generation sequencing on our metastatic colorectal cancer cases, because when we do NGS, that's the only way that we can actually determine whether they are TMB high versus not. Whereas for dMMR, we can actually do the immunohistochemical test. So that's my comment. And the question for you, Dr. Marwa.
Okay. Thank you, Dr. Kanaan, for your question. This is an interesting question. The first thing came to my mind is that a relationship between the gut microbiome and the hepatocellular carcinoma? So this is the first question that I would like to ask myself and look at the literature to see first if there is a relationship or if there is an evidence of the gut microbiome and the hepatocellular carcinoma. What I know is that the gut microbiome regulates immune system, educate the immune system how to function well. So it might be there is an indirect relationship between the gut microbiome and the hepatocellular carcinoma. Now for the antibiotics, there is an evidence and there is an agreement in the scientific world, especially in the microbiome, that the antibiotics influence negatively the gut microbiome. So the antibiotics influence the function of the gut microbiome, induce a dysbiotic state in which there is imbalance between the harmful and beneficial bacteria. So it might be influenced indirectly that the patients who is receiving antibiotics have their dysbiotic state in their gastrointestinal tract, and then indirectly the gut microbiome might influence the prognosis of hepatocellular carcinoma. Yeah, this is what I would like. Thank you.
Thank you, Dr. Khan and Dr. Marwa. I would answer the question in some facts rather than making the decisions. I'm not going to say what is right or what is wrong. The first thing, I read a few papers saying that the samples that turn to be MSI high, but they are TMP low, when they repeated the experiment with different methods, they were in fact not MSI high. It was just a technical problem. And then some other papers showed that just like the one I presented, those patients are in fact not responsive to immunotherapies. The third fact is that several groups are trying to change the idea of doing the DMMR using the immunohistochemistry and just use the mismatch repair or MSI with PCR. Instead of doing PCR, you try to use the panel, the same NGS panel that can involve the or give us an indication about the MSI. With doing that, it will be financially less expensive. But I know after all that, there are some groups, and the first fact is that even if the DMMR, even if there's no deficiency in the proteins, it has been shown that in some cases, there is somehow that the DNA damage is still happening and that there's still microsatellite instability, even if those proteins are expressed by IHC. It could be epigenetic modification. So even if you do IHC, it wouldn't be the best way. So all these facts highlight that we should, or maybe it could be better to use the margenomic instead of the proteomic. I know that some people would disagree. So I think, and we don't have an exact data to show us if we should not use the MMR. I know it's cheaper, and it's one of the reasons why we do it here. Thank you.
I have a question for Dr. Sharif. So my question to Dr. Sharif is, you were talking about clinical trials and the use of molecular profiling. However, just to be realistic, what do you think we should do in retrospective fashion? Should we fish for a specific gene or test, or should we go with a whole exome or NGS? What's your recommendation? And you can give example of the major cancers in Saudi Arabia, like breast and colon.
Thank you very much, Dr. Marwan, for this question. Actually, you know, when we are talking about precision medicine, the best scenario is to test any biomarker in a prospective way, but this is not always easy. It needs infrastructure, it needs a well-dedicated clinical trial team, it needs a networking. It's not always easy to do, especially here in Saudi. So we were going in a retrospective way. Again, it depends on the facility. It depends on the how heterogeneous is expected the tumor subtype. So, for example, I'm telling you in a disease like lung cancer, my preference is to go with more sophisticated techniques like NGS or whole exome analysis. Because you have expected to have a lot of small subgroups of patients, a small subgroup of biomarkers, and you can correlate that with the outcome of interest. In a disease like breast cancer, we don't have clear evidence of the activity of many subgroups, of many small subgroup of biomarkers in a subset of patients. So we need to take that with caution. However, by the end of the day, whatever the technique you are going to choose or whatever the biomarkers they are looking for, this need to be taken with caution. Of course, it will be a kind of retrospective analysis and you need to validate again in a prospective way. The other point that I need to have just a small comment of trials that have presented in the presentation of Dr. Kanaan. He presented data of clinical trial later lines of treatment like Shiva, Mosquito, and so. In Shiva, for example, if you look to the PFS of the group of patients who have been treated based on the biomarkers, it is not that great. PFS 2.6 months or so, and it is not far away from patients treated irrespective of the biomarkers. So again, without clear validation of the biomarker, it seemed that it's still early to make a treatment decision based on a biomarker that's not validated to have a clinical impact. So it needs to be taken again with caution. And that's why we have the classification released by ESMO, SCAT classification, tier 1, 2, 3, and 4. And they are recommended to use within the clinical practice of patients having biomarkers within the class 1 or class 2. But patients in class 3 and 4, that's to say patients having only data from some small series of patients or preclinical data, you don't need to use these biomarkers or the results of these biomarkers in decision-making in routine practice outside clinical trial. So this is my answer. Thank you.
Thank you, Dr. Sharif. I just have a very small comment. I hope you can hear me. Can you hear me?
I can hear you.
Right.
Yes.
So Shiva is one of the oldest molecularly targeted precision oncology platforms, and it dates back to 2013. In fact, it was published in 2015. And we know that biomarkers back in the day were not as abundant as biomarkers now. طبعا impact and Memorial Sloan Kettering impact, and there was actually a big publication by Moore's Cancer Center. I actually have the data, and they have shown that when you give molecularly targeted therapies to pre-treated patients, that progression-free survival and overall survival is actually superior. So I thank you for mentioning Shiva, but it's one of the older trials.
Of course, I think so. Before going for using a medication, which for a biomarker that's not well validated, as you mentioned before, this needs to be discussed within a molecular tumor board. You need to have someone experienced for that, and we need to take caution because we have examples for certain biomarkers that's working well with the medication in certain tumor types, but not working in the same way in another tumor type. The clear example, or the most famous example, is BRAF mutation. We have clear activity in melanoma, but single agent BRAF inhibitor are not working in colorectal cancer. It needs to be combined with another agent. So it is a complex way. It needs some knowledge about the microenvironment and molecular background of each tumor type before making decision about use or not using the biomarker, which is not well validated in previous times.
Thank you so much. We have one more last question. Usually patients go under screening if they are before they receive PD-L1 blockade. And despite if they do have expression of PD-L1 or not, usually they do respond to the treatment. Do you think that maybe in the future it's better to adapt additional screening in addition to the tumor screening, maybe in the blood, to check if there is an immune cells that do express the PD-L1 despite the expression on the tumor itself? And this is also because usually the patient do receive the treatment in the IV, so in the blood. What do you think of that? What's your comment?
I would just say that exactly what everyone is trying to do is to find a biomarker. What should we do from the screening as a predictive biomarker?
So I think if someone finds a excellent biomarker for immunotherapy, and this would be, and as they say, the holy grail of oncology, I think we still lack the most appropriate biomarker. And tumor mutational burden being high, there is evidence of TMB more than 10 in, let's say, melanoma, that they actually don't respond to immunotherapy compared to other cutoffs. In fact, one publication said that you have to have a TMB of more than 44 mutations per megabase to respond to immunotherapy in melanoma. Any biomarkers, I think it's a huge field and it's a developing field. PD-L1 expression, I think, differs between even disease sites. Now we have CPS scores, we have TPS scores, which are a combination of immune cell expression plus tumor cell expression. And I can see a distinguished pathologist here with us who I'm sure is going to talk about this specifically tomorrow. So this is a developing field. Immune expression and tumor expression is not the same. However, I think drug companies mainly who are developing biomarkers to basically enrich for populations that may respond to immunotherapy are developing several calculations that may give us a hint on who is going to respond versus who's not going to respond.
Thank you so much.
Can I have
Yes,
a
yes.
comment on that, please?
Absolutely.
Just I will add something to what you have mentioned, Dr. Kanan. In breast cancer, triple negative breast cancer, I'm doing breast, So basically, those patients were selected for immunotherapy trial based on PD-L1. Okay. So in the Ambition 130, and in addition to the PIMPO trial, the selection at the trial was based on the PD-L1. And they found that the hazard ratio for progression-free survival based on PD-L1 selection in the Atezo trial, it was 0.65, if I remember well. They did later on retrospective analysis using some NGS-based biomarker, and you know that in triple negative we have certain molecular classification based on biomarkers. The patients were classified into one of these classifications: basal-like immune activated, basal-like immune suppressed, mesenchymal, and so on. And in the category of basal-like immune activated, that's to clarify it, it is expected to be the immunogenic category within triple negative breast cancer. When they looked for the activity of Atezo, nab-paclitaxel, selected by basal-like immune active subtype in this classification, the hazard ratio came to be 0.51. So this category of patient, based on this new biomarker, it seems to have more benefit from including immune therapy compared to selection based on PD-L1. However, it is 27% of patients compared to 40% of patients with PD-L1 positive. So we know that in some tumor types, what I want to say, adding to what you have mentioned, we know that there is certain fraction of patients who are more likely to respond to immune therapy. But the beauty is how to select those patients. PD-L1 is a biomarker. It is not the perfect one. It is not the one who could predict the highest benefit from immune therapy, but it is the only one validated in clinical trials in many tumor subtypes. But do we have more perfect biomarkers? It seems so. Basal-like immune active in triple negative breast cancer need to be activated in prospective fashion. In colorectal cancer, in gastric cancer, we have benefit from immune therapy plus chemo in PD-L1 positive patients. However, in subgroup of patients in clinical trials, we have more benefits in the small subgroup who have MSI high tumors. There seem to be some overlap. So the best biomarker up till now need to be checked. PD-L1 is one of them, but it's not the best, of course.
Okay, thank you so much. With no further questions, we will end our first session. We're going to take a break for 15 minutes, and then we're going to be back for our second session.
So we will start at 4:20, inshallah.
4:20, inshallah.
Thank you very much. Excellent. You're doing a very interesting study.
Mic test. 1, 2, 3. Mic test. 1, 2, 3. Mic test. 1, 2, 3.
Fahad. Assalamu alaikum.
Okay, so Bismillah ar-Rahman ar-Rahim. Welcome back to the second session for the day. It will be about molecular tumor board and cancer genetics. And it's my pleasure to invite our moderator for the session, starting with Dr. Shawqi Bazarbashi. He's a senior consultant medical oncologist from King Faisal Specialist Hospital and Research Center. And we have Dr. Fahad bin Shamsa, chairman of oncology at King Fahad Specialist Hospital in Dammam. And we have Dr. Fahad, the Migabilla consultant of medical oncology at King Faisal Specialist Hospital and Research Center. So the floor for you.
Okay, so maybe I can start, Fahad?
Yes, please go ahead, Dr. Shawqi.
So I think we have an excellent program today. Thank you for giving me the chance to moderate with my colleagues, Fahad and Fahad, this interesting part of the symposium, and I hope that we can all learn from your experience. So I understand there has been some changes in the flow of the lectures. The first presentation will be by Dr. Kanaan al-Shammari. It will be a local experience of molecular tumor board in National Guard Hospital. And I would like to call for Dr. Kanaan. Dr. Kanaan is a consultant medical oncologist at King Abdulaziz Medical City National Guard Ministry. He's also the chair of the NCCN chapter for the MENA region, and he is the director. And so we will be looking for his presentation. Kanaan, the floor is yours.
Thank you very much, Dr. Shawqi, for the introduction. So this part of our talk will mainly touch upon precision oncology applications, or at least one of them, which is the molecular tumor board, our local experience at National Guard in Riyadh. So what are molecular tumor boards? They're basically specialized tumor boards that study the complex genomic profile of patients and recommends therapies based on the results of the comprehensive genomic profiling of patients. Of course, one of the prerequisites for efficient molecular tumor board is to have an extensive next generation sequencing panel that is validated, that is accurate, and can provide you with solid results. Now, we have chosen in National Guard a commercially available next generation sequencing test that involves sending a tissue to a central lab and performing the test, and then relaying the results to the treating physician, who then needs to review, understand, and decide how to use the results to benefit the patient. We basically chose an FDA-approved NGS that tests more than 300 genes. And the test is basically Foundation 1 CDX. So this is one of the earlier FDA-approved tests that assesses 324 cancer-related genes. It does report MSI and TMB, and is indicated for at least 7 cancers. And it does provide, or it is approved for use for more than 25 FDA-approved targeted therapies. So molecular tumor boards are critical in precision oncology programs, because when we are faced with a complex profile, it may be challenging to interpret and may be challenging to choose non-FDA approved drugs. So MTBs do discuss patient characteristics and information. They also discuss complex genomic or molecular information compiled with pathologic data that is reported by in-house pathologists And then after looking at all of these pieces of information, they recommend treatment options and give us corresponding evidence levels. They are a critical hub for patient management. They facilitate interpretation of complex molecular data. They assign targeted therapies to patients who may benefit, depending on the evidence level. They provide education, and this is important for us as physicians who would like to practice precision oncology. They give education on molecular pathways, molecular assays, and how these relate to therapy strategies. They describe complex genomic pathways and how to target those pathways. They keep track of clinical trials and they screen for clinical trials. Now, this is more applicable in the United States and Europe. So MTBs, now I chose this funnel-shaped graph here or picture. When you have your sequence data, this is processed and in an automated fashion, and then you get event detection. This is followed by filtering, reviewing, and validating these results. Then you get functional prediction. Then this piece of information gets given to a genomicist who has expertise in interpreting these complex reports, and then they would produce the genomic report. And then all of this information is put on a report to the treating physician, where they need to think about how to clinically apply this piece of information. So MTBs have pathologists, have scientists who have expertise in genomic pathways, have oncologists, should have data management experts, because you are faced with large amount of data, radiologists, and other healthcare providers. As mentioned, in the MTBs, you get help in interpreting results and recommendation of best available therapeutic options. Most large Western cancer centers have their own molecular tumor boards. Most famous and probably the most successful example is the Memorial Sloan Kettering MTB with their IMPACT study. Now, this is a large prospective study that actually continues to find new genomic biomarkers and continues to actually create early phase clinical trials based on what they find. Now, interestingly, their in-house MSK IMPACT NGS tests for almost 500 genes. So let's talk about what we've been doing in National Guard Riyadh. So we started as a small initiative. We started formally in late 2020. Now, if you remember, these were the horrendous COVID times. However, we were determined to start. We had molecular tumor board meetings that occur monthly, plus minus an ad hoc meeting depending on clinical need. Between 2 to 4 cases get presented and discussed over an hour via a virtual platform. We always have a scientist or a genomicist, an oncologist, and of course the oncology main responsible physician. This is the minimum requirement of personnel that are present. Other interested physicians from other centers have also attended, and we have always had our pathologists and our scientists attend. Now, this is, I have to say, in collaboration with Roche, and this is the Foundation One Molecular Tumor Board. So what's the goal of actually doing a molecular tumor board? The goal is to give access to cancer patients to a quality controlled CGP, which we have, and the ability to be discussed in a scientific, molecular, high caliber tumor board. Secondary goal is to expand this to becoming a national MTB that will benefit patients from other areas of the kingdom. So let me tell you who have we presented and how and what are our results. So we chose to actually present patients after having had the expanded panel NGS, which is Foundation One, and have had a complex genomic profile, meaning if the patient comes with an NGS profile showing a ROS1 alteration, and they are a non-small cell lung cancer patient, this is a tier 1 mutation. Standard of care is to actually offer a TKI against ROS1. So these patients do not get presented. So we present non-tier 1 mutations and no known or FDA approved or tier 1 recommendation for a therapy, and no known targetable mutations. Patients who we select to perform the expanded panel NGS on and present them in the MTB are chemorefractory patients and who are, in the judgment of their treating oncologist, have a prognosis of at least 6 months. Now, why did we choose this 6-month figure? It's basically for a few reasons. The first reason is the test takes time until the results are back. Now, there's always a promise that it comes in less than 4 weeks, which is a kept promise. However, if we order a non-formulary medication, we have to actually account for the lag or the time lag that the non-formulary medication would arrive. Now, most of these cases have actually come from National Guard Riyadh. However, we have successfully included oncologists from outside of our institution. Now, what I'm showing here is just an example of 2 Foundation 1 reports. One of them is for thyroid carcinoma, and it does show that the finding in the genomic profile is a TET2 mutation. Now, as medical oncologist, and I can speak probably for myself, Dr. Mohammed, Dr. Fahad, and Dr. Shawqi, that we do not know a medication that would target TET2. However, discussion with the genomicist did explain that it does affect your cancer immunity, and perhaps maybe immune checkpoint inhibitors may have a role here. The other case is a case that I actually treated of a elderly gentleman with gallbladder adenocarcinoma who unfortunately progressed on 3 lines of systemic therapies. Now, the Foundation 1 report showed this patient to have a HER2 amplification. Now, we know that HER2 is not a regularly tested biomarker in gallbladder adenocarcinoma. And then hence here, the report does not recommend therapies. It does recommend therapies based on other tumor types. Now, I did give this patient anti-HER2 therapy, and the patient actually stayed on therapy for 8 months. So who are the cases that we have presented so far? Well, I think most of my colleague oncologists know that I have an interest and specialize in sarcoma. So automatically, the highest number of patients that have been presented are actually my soft tissue sarcoma cases. This is followed by pancreatobiliary and liver cancer, so biliary tract cancers, followed by head and neck cancers, followed by upper and lower GI cancers, followed by skin cancers. So this is our preliminary results. Now, I actually looked at this last month, and I did a quick analysis based on the cases that we have presented so far. So median number of prior lines were 2. Mean was actually 2.5 lines. Now, the range of the lines was actually 1 to 8, meaning one of the patients that we actually presented progressed on 8 lines of systemic therapies. So I guess what I'm trying to say here is that this is a cohort of patients that have been heavily pretreated. Now, 94% of patients that were presented in this tumor board had recommendations of therapies. 73% of all patients presented in our molecular tumor board had recommended therapies based on their genomic profile. So had genomic recommended therapies. So this equates to 3 out of 4 patients having had recommendedly genomic therapies recommended. Now, I have to say, disappointingly, less than 50% of the patients that we actually presented, we were able to actually give them genomic therapies. Now, one of the questions that I'm sure is going to come up, so I'm going to go ahead and answer the question before it comes out.
It's very good, 50%. Yeah.
Well, I was hoping for more, Dr. Fahad. And why do we only give 50% of our patients? And there are multiple reasons. One of the reasons is that these are heavily pretreated patients, so their prognosis is poor. So actually, the majority of these patients actually passed away or died before being given targeted or genomic-based therapies. Some of them, actually very few of them, in fact, just one, I was expecting the number to be higher, were waiting for a non-formulary drug to arrive to be given this medication. Some of these patients are actually still responding on prior lines. Like the outcomes of our molecular tumor board, and to be honest, this is what I'm proud of. If you look at the second statement, so 50% of our patients who got given genomic therapies remained progression-free for 6 months at least or above, giving a median progression-free survival for our molecularly targeted or patients who are presented in our molecular tumor board who are chemorefractory patients. Their median progression-free survival, I can proudly say, is actually 6 months. So this is what precision oncology is all about, and this is what touches patients, and this is what impacts on patient journey and patient care. I'll give a few case examples. So this is a 23-year-old man who I treated in my clinic a couple of years ago. He came to me from the States with a diagnosis of metastatic hepatoblastoma. He was heavily pretreated. In fact, when he came to me, he had been given 3 prior lines of therapy. He had an opinion from the United States that gave him a prognosis of 3 months. I gave him chemotherapy, systemic chemotherapy, after consulting a pediatric oncologist because hepatoblastoma mainly affects the pediatric population. So I gave him 5 different types of chemotherapies given in an alternating manner, and he progressed. So we performed Foundation 1, and he was found to have a CDKN2A/B gene mutation. So I sat with the patient and his family and explained to him, we have 2 options, either best supportive care or an experimental palbociclib, and he chose to be given palbociclib. Now, palbociclib for my non-oncology colleagues is FDA approved for breast cancer. He continued to respond for almost 2 years. Another case is also a patient that I treat. She's an elderly woman with metastatic leiomyosarcoma originating from the liver, metastases to lungs. She was heavily pretreated. I gave her in my clinic 5 prior lines. So doxorubicin ifosfamide, which is standard of care, gemcitabine docetaxel, pazopanib, dacarbazine, and trabectedin. All of these are FDA approved drugs in leiomyosarcoma. We performed NGS, and it did show a JAK3 mutation. We know that JAK3 mutations do not have a specific targeted drug that targets the JAK3 mutation. She also had ATRX and RB1 loss. We discussed it in our molecular tumor board, and there was discussion that perhaps JAK3 mutations can upregulate PD-1, PD-L1 expression, and hence immune checkpoint inhibitors had some sort of a recommendation. So I gave her nivolumab and ipilimumab, and she remained progression-free for 6 months, and she is still alive. Another man, I think this is your patient, Dr. Mohammed, who had metastatic colorectal cancer. He progressed on the standard 3 lines of cytotoxic chemotherapy. He had no further evidence-based lines. His CGP showed a tumor mutational burden of 14. Now, TMB is not a regularly tested biomarker in colorectal cancer, and he also had the KRAS G12C mutation. He was given pembrolizumab. He did well for 16 months. He unfortunately died 3 months after stopping pembrolizumab. Now, this was prior to the time of the FDA approval of the G12C inhibitor that is FDA approved in lung cancer. So we were not able to access the drug in a timely fashion. Another example of perhaps a group that are doing something similar to ours is a German group. Basically, they call it a master protocol. It's a prospective observational precision oncology program where they sequenced almost 1,500 patients. 1,300 patients were actually discussed in their molecular tumor board. Interestingly, and I suspect that this 1,200 or 1,100 patients probably equate to 80% had therapy recommended by this MTB. And I'm not sure what 40 out of 1,200, probably 1 out of 4 patients had therapy given. Now, the reasons for non-implementation of the molecular tumor board decisions were similar to ours, I have to say: worsening of patient's general condition, death before treatment initiation, and lack of access or reimbursement of the recommended drugs. So how did they do in terms of patient outcomes? So there was an improvement in the objective response rate compared to the last or the previous systemic therapy by a modest number, so 8%. There was a 9% improvement in disease control rate compared to the last systemic therapy. And 1/3 of patients had a progression-free survival ratio of more than 1.3. Now, this ratio means progression-free survival of the study drug compared to the progression-free survival of the previous line. And if the result is more than 1.3, it means that you get a 30% benefit. So 1/3 of patients actually benefited in terms of progression-free survival. Another example was done by the University of Kentucky. They did twice monthly MTBs, so similar to ours. They did virtual MTBs. And the reason for doing those is overcoming challenges of insufficient local infrastructure or expertise. Now, interestingly, these results show only their non-small cell lung cancer cases. And if you look at the survival curves, patients who have been presented in their MTB survived longer compared to patients who have not been presented in their molecular tumor board. And these are non-small cell lung cancer patients. So to recap, comprehensive genomic profiling reports give us a detailed description of patients' genomic profile and an overview of therapeutic options. Molecular tumor boards are the next generation tumor boards. They do require a considerable amount of effort to establish and maintain. I think I have shown you that they have improved patient cancer or cancer-related patient outcomes. They would improve enrollment into genomic-based clinical trials or biomarker-based clinical trials. They are becoming a key pillar in the potential or realizing the potential of precision oncology. Some of the potential challenges that we face and other programs face is that we may be unable to get the targeted precision oncology drug in a timely fashion. The timing of NGS. Now, I mentioned that less than 50% of patients were actually exposed to or given the genomic-based therapy. So timing of NGS, are we actually doing it later in the patient cancer journey when they become resistant and sick, or should we do it early in their therapy journey? There's also difficulty in engaging with other centers, mainly due to their lack of in-house NGS or their external NGS technology. So the take-home messages from this talk is that NGS is standard of care in certain cancers, with indications expanding rapidly. Molecular tumor board examples are successful in providing patients with genomically based therapies and access to genomic or biomarker-based clinical trials. Now, we know that the historical survival for chemorefractory patients or post-exhaustion of standard of care therapy is less or equal to 3 months. Yet I have shown you that 50% of our chemorefractory patients continue to respond on genomically recommended therapies for 6 months or longer, based on the discussion in our molecular tumor board. So I think this is what precision oncology is all about. So molecular tumor boards have improved cancer-related patient outcomes. And by this, I conclude. Thank you.
Thank you, Dr. Kanaan. I think for this excellent presentation, we will proceed with the agenda, and then we will leave the discussion to the question and answer session. So I'll give the microphone to Dr. Fahad bin Shamsa.
Thank you, Dr. Shawqi. Thank you, Kanaan, for this very interesting and special experience presentation that raised a lot of questions. So we will have a chance to discuss it. But now we want to take it to some practical setting where we have the live molecular tumor board cases. We will be joined by an expert from USA, and I will give you the mic to moderate this session, Kanaan. Thank you.
Thank you, Dr. Fahad. So I'd like to take this opportunity and thank our partners, Dr. Saad Khan and Dr. Umar Hamdani. Dr. Saad Khan is an assistant professor and a consultant medical oncologist who specializes in head and neck cancers. He has been our partner in our molecular tumor board for the last probably year and a half or so. And I also would like to welcome Dr. Umar Hamdani, who's an expert genomicist who is going to help us in our molecular tumor board today. So we are going to present two cases. One is a case that was treated by my colleague, actually, Dr. Samah Al-Rahili from National Guard, Jeddah. She is luckily on holiday, so she gave me this task of actually presenting her case. And the other one is a case that was treated by my colleague, Dr. Mohammed Al-Garni. So we're going to talk about the cases first, presenting the history, and then we're going to give the microphone to Dr. Umar Hamdani, who's our genomicist, to discuss the Foundation 1 report. We're going to hear from our expert, Dr. Saad Khan, who is going to talk about therapeutic options. And then the presenter of the case is going to talk about the subsequent patient journey. So I'll start with case number 1. So again, this is a case that was treated by my colleague. So she was referred, a 28-year-old woman who was diagnosed with a pseudopapillary tumor of the pancreas. She was referred to a National Guard hospital in Jeddah for a second opinion. When her initial diagnosis occurred at 24 years of age, where she presented in December of 2017 with an abdominal mass and core biopsy reporting extrapancreatic solid pseudopapillary neoplasm. The scans showed a large mass at the gastrohepatic space stretching the pylorus. She was discussed at a local tumor board, and they decided for surgical resection. This occurred in early 2018. She recurred, unfortunately, a year later, and she was given multiple lines of chemotherapy. So she was given cisplatin and gemcitabine with disease progression until March of 2020. This was followed by irinotecan. Now, this occurred during the challenges of COVID-19, where 5FU, I think, was short. She progressed in August of 2020. And then in the third line setting, she was actually given Zelox, where she actually also progressed after 3 cycles. Now, this dates back to November of 2020. She was seen at National Guard Jeddah. Her performance status was 2. However, she was still young, in 26 or 28 years of age. Prior pathology slides were requested to be reviewed by their in-house pathologists, and Foundation 1 was requested. Now, this really shows you how extensive her disease burden is. So she had extensive liver metastases. She had significant hepatomegaly. She had right hepatic lobe pseudoaneurysms, and she had para-aortic lymph node enlargement. She also had ascites, so she was decompensating, in fact. Now, the local pathology report from the hospital basically confirmed the finding of a solid pseudopapillary tumor of the pancreas. However, IHC, which was done by the pathologist there, showed the cancer cells being positive for PR, so progesterone receptor. Now, this is the Foundation 1 report, and I think I will leave this now for you, Dr. Umar.
Sure. Thanks for having me, everyone. Just to comment on what was said earlier about how basically tumor board-driven therapies can increase matching to a therapy that might be actually more beneficial for the patient. I would urge everybody to check out Roselle Kurzrock and Shumei Kato's work on N of 1 studies. They really are kind of doing the same thing, and they've taken the same route where they can really show a clinical benefit by using these types of MTB guided therapies. But getting to these genomics, I just want to touch briefly first on the tumor fraction here, because especially in this tumor type, pancreas solid pseudopapillary tumor, we only have 39 in the FMI core database, and we have over 500,000 cancer types in our database, or rather 500,000 cancer reports in our database. So with only 39, the statistical power to tell you about the frequencies isn't going to be too useful, but I just wanted to mention that TF, at least, has been shown to be preliminarily a biomarker in non-small cell lung cancer, which may have a prognostic effect on the response to therapy. So we might actually be able to use it in the future to tell how much therapy we should actually give a patient. So do they need mono IO versus chemo IO? And that can be kind of extrapolated from ctDNA positivity from this tumor fraction estimate. So on our test, tumor fraction is basically going to be reported these days, or at least in the current state as our test form is, as tumor fraction elevated or tumor fraction not elevated. In this case, you would get, if it was to a tumor fraction value of 18%, it would have been displayed on our new reports as tumor fraction elevated as well. But moving on to the actual alterations here, you can see there's an NF1 alteration and there's a CTN and a B1 alteration. So if we can move to the next slide. One more slide, actually. Great. So this is a schematic of the mTOR and MAPK pathway. You can see to the left there, NF1 is just below that kind of double arrow diagram of ROS GTP and ROS GDP. And basically what NF1 is responsible for doing is removing, or rather hydrolyzing GTP that's attached to ROS and pushing it towards this ROS GDP inactive form. So when you lose NF1 function, technically ROS is remaining in its active form longer and the pathway could be hyperactivated. Preclinically, that has been shown quite well. And then as well, you can see here, which will be a little bit important on our next, or when for Dr. Khan's data, is that ROS has this tangential interaction with the mTOR pathway, and that there's feedback loops that kind of occur between them and boost the signal of both of those pathways, because they are both major proliferative pathways and kind of essential to cancer. So if we can move to the next slide. The other thing I'll say about NF1 alterations too is that I looked in our database, and out of the 39 reports, there actually was no reports of an NF1 alteration in this cancer type. So that was interesting to see, just because NF1 is usually somewhat common. But in pancreas carcinoma of no origin specified, NF1 short variants are present in about 3% of the 2,500 reports we have. So take that as you will. I did read some reports that say that this type of cancer makes up maybe about 1% of pancreas cancer, so it is a very rare cancer type. But I also wanted to talk about the beta-catenin alteration we have here. So this specific alteration, CTNNB1 S37F, has been seen in 7 out of 39 pancreas solid pseudopapillary tumors, which is pretty striking, almost a fourth. But this specific alteration is very important too, because beta-catenin has both tumor suppressor gene and oncogene functions. And this specific alteration is an activating alteration that basically leads to an oncogenic scenario, making this a driver alteration for beta-catenin. And that presents a very interesting clinical option for the patient in that, and I won't get too much into it because Dr. Khan will be talking about it more, but there are several interactions between the Wnt pathway and the mTOR pathway, in that the Wnt pathway turns on the mTOR pathway, or rather activates the mTOR pathway, and as well the mTOR pathway then inhibits inhibitors of the Wnt pathway. So there's this kind of feed forward or feedback cascade, feedback loop that happens when both of these pathways are, or when one of these pathways are activated. And here you actually have what could be an activation of both of those pathways. So with that, I will pass it off to Dr. Khan.
Thanks, Dr. Hamdani and Dr. Kanan for that nice introduction. So as a practicing medical oncologist, of course, it's very difficult when someone presents a case of pseudopapillary neoplasm of the pancreas, because when I was in fellowship, I never saw a case like that. And then I'm given a foundation medicine report or other molecular genetics report that says CTNNB1, and I actually don't know what that means until I meet with someone like Dr. Hamdani, who just explained that it can interact with the mTOR pathway. So with that initial piece of information, I can go into the medical literature and hear from JCO Precision Oncology. We were fortunate that even though this is such a rare tumor, there were data suggesting that there is activity in patients that have pseudopapillary neoplasm with the neoplasm of the pancreas with activity of everolimus and those who have CTNNB1 and PTEN abnormalities. Now, again, it doesn't perfectly match the patient that we have the genetic report for, but it gives us an idea when we're running out of other options that perhaps we may be able to target the mTOR pathway. And Dr. Hamdani, the genomicist, gives us information that, oh, perhaps mTOR may be active because of the molecular crosstalk between mTOR and CTNNB1. And I use everolimus quite regularly in my other patients. So I say, okay, well, let's see if that might be reasonable for the practicing oncologist who's referring this case to the molecular tumor board to try. And ultimately, the decision goes back to Dr. Al-Shabari, and I'll leave it up to him to continue the conversation. Oh, just quickly, it also can tell us what not to use. So in this case, CTNNB1 is associated with perhaps less response to immune checkpoint inhibitors. And that's one of the additional benefits of molecular therapy, that the more you understand about the genetics of the tumor, you can find out what might work, such as mTOR inhibitor, and what is less likely to work, such as immune checkpoint inhibitors. And now I'll go back to Dr. Al-Shabari.
Thank you, Dr. Khan. So back to our case. So the treating oncologists actually decided to give this young lady tamoxifen based on immunohistochemistry showing PR positivity. Now, this was prior to the molecular tumor board discussion. So she started tamoxifen. There was decent tolerance of the medication. And then after the MTB discussion, and after what Dr. Hamdani and Dr. Khan explained about perhaps mTOR inhibition may be of value in this case, the treating oncologist decided to start everolimus. Symptomatically, the lady was actually improving. And radiologically, I apologize about the long report. However, the conclusion of this is that there was a significant partial response of this patient. So she did respond radiologically and clinically beautifully. And this shows you how significant of a reduction in her liver metastases have happened. Now, the patient still remains on therapy. She has had a progression, or she remained progression-free now for at least 8 months. Now, remember, she started with decompensated liver disease, with ascites, very large liver metastases. Her performance status now is 0. Now I leave the second case for Dr. Mohammed again.
Thank you, Dr. Kanaan. So my case is less complex than yours. This is a 33-year-old woman. Actually, now she's a 37-year-old woman. She was referred late in 2019 after being investigated in another hospital for shortness of breath. She was found to have lung lesion and mediastinal lymph nodes, and the biopsy at that time revealed high-grade carcinoma, more of a small cell carcinoma with some neuroendocrine feature. She had also metastasis at that time to the breast, pancreas, and lung. So she was treated as a high-grade small cell, high-grade neuroendocrine tumor, and she was given cisplatin etoposide with stability of disease, something that you would expect to see some response in patient with small cell cancer. Then after she progressed, she was discussed on the thoracic tumor board, and the consensus was to proceed with second-line immunotherapy. So she was given pembrolizumab with mixed response. Then she had multiple lesions that was painful and subcutaneous, and some bone lesions that was treated with radiotherapy. And then she had more of disease progression. And at that time, given the unusual course in terms of lack of any response to platinum-based chemotherapy, we decided to repeat the biopsy for pathology review again and for molecular study. And her pathology revealed a neuroendocrine tumor with a Ki-67 of 30%. And at that time, we requested for foundation one. And now I will leave the microphone to Dr. Hamdani.
Thanks. Yeah, so there's not really a reason to show front page genomics here because there was really only one alteration found in this report. But the TMB on this report was 8, in CUP. That's certainly an investigative biomarker. And the fact that you've used immune checkpoint inhibitors in the second line, there's at least some decent data in cancer of unknown primary to show maybe 20% of cancer of unknown primaries do respond to something like an immune checkpoint inhibitor. And those numbers do change a bit depending on what biomarkers you're looking at, TMB versus PD-L1. Well, moving on to this ROS1 oncogenic pathway. This is a very similar diagram in a sense to what I showed you for the MAPK and mTOR pathway schematic that was in the last case, and that most receptor tyrosine kinases, ROS being a receptor tyrosine kinase, are transmembrane proteins that will activate downstream signaling pathways such as the MAPK pathway and the mTOR pathway. And one important thing about ROS1, as well as ALK, NTRK, any of these other receptor tyrosine kinases that are prone to fusions in some frequency, is that if you have a fusion that results in the kinase domain being fused to an active promoter, it basically creates a ROS1 fusion protein that's free inside intracellularly and able to activate all of its targets. At least predicted to be able to activate all of its targets. So just to give you some numbers on ROS1, if this was something like an FGFR fusion, it does have more diagnostic capability in that they are overrepresented in some diseases, specifically things like cholangio and urothelial carcinoma. But ROS1, actually, just looking at our database broadly, I just looked at the top frequencies for ROS1 rearrangements. And basically what I've found is that soft tissue liposarcomas are the top with 1.4% of them having a ROS1 fusion. And lung adenocarcinoma is second with 1% of those having a ROS1 fusion, or rather a ROS1 rearrangement as our database lists it. So it's really not something that you're going to find in high prevalence. That's basically what I wanted to say. So it's looking at these things in a pan-tumor sense is certainly something worth doing, considering that the trials for things like ROS1, some of the ALK inhibitors, NTRK, were done pan-tumor, and it might very well likely be because of the frequency we find these alterations at. We can go to the next slide. This is basically a schematic showing, first off, the wild-type ROS1 protein at the top there, and its genomic structure, and then below some common ROS1 rearrangements. So here we've detected a CDC30 ROS1 rearrangement. What's really interesting about this is that I only found 3 reports out of, like I said, over 500,000 reports in our database, and they were all in thyroid papillary carcinoma. And then as well, just from a literature review I did, I also found reports of this at least twice in thyroid cancer as well. So take that as you will. That's literally an N of 5. So it's really not something you could use frequency wise, but that is some decent information because we haven't observed it elsewhere. As well, this specific fusion picked up CDC30 exons 1 through 10, which would have the promoter in it, and ROS1 35 or exons 35 through 43. And as you can see from the top of from the schematic at the top here, that's basically exactly the tyrosine kinase domain of ROS1. So what's predicted to be an activating fusion here, because it is the kinase domain of ROS1 potentially expressed under an active promoter. And there is a host of ROS1 inhibitors out there, but I will not steal too much of Dr. Khan's thunder.
Right. And then just to Dr. Hamdani's prior comment, this slide is from Dr. Cato and Dr. Kurzrock, who are my colleagues on the Pacific Coast at University of California, San Diego. I was reminded while we were discussing this case when I was doing my fellowship in the early 2000s, the problem that the oncologist faced was that we would say we only have 5 drugs that we can give our patients with thoracic cancers or carcinomas of unclear primaries. And then we would run out of all the options. today, the practicing oncologists, certainly those that use molecular tumors, molecular characterization of tumors, the problem is alternate. The problem is keeping track of all of the different mutations that are potentially actionable. So for example, for me as a practicing oncologist at Stanford, I know that ROS1, NTRK, RET, all of these are very standard agents that have essentially pan-tumor FDA-indicated therapies that we can give. So a oncologist down the street who may not be as specialized in the treatment of one particular cancer may have a harder time keeping track of all of the different drugs that are out there and available and potentially matched to a mutation. And so a graph like this or schematic like this is helpful, where if you have a patient who undergoes molecular profiling for a tumor of unknown primary, depending upon whether they have a HER2 amplification like Dr. Kanan's earlier case did, or an EGFR alteration, it reminds you to think about that. I will point out that even though this is not a very old schematic, this actually does not include ROS1. So we can proceed to the next slide and show what impact does next generation sequencing have on patients with carcinoma of unknown primary at a large cancer center, in this case on the left. This is the experience of Moffitt Cancer Center in Philadelphia, in Florida, where they looked at roughly about 50 patients that had molecular profiling performed, and the area in light blue shows that there was an indication established for immune therapy. And the area in darker blue means that these were patients that were identified as having a molecular driver that was then actionable. So it's certainly for cases like this that are neuroendocrine tumors or extrapulmonary small cell tumor. In the United States, in my practice at Stanford, of course, everybody would be getting molecular profiling at the time of diagnosis, because there is so much actionability built into it. But again, the circumstances are different in California versus where you may practice. And then on the right, this is just a similar schematic to what Dr. Hamdani mentioned, that ROS1 is a well-established fusion partner, and rearrangements result in increased cell survival and proliferation. So if we proceed to the next slide, we can show what that means for patients. And at the top is just a very simple announcement that basically says that in addition to crizotinib, which was used in this case, in the United States, a drug called entrectinib is also approved for use in ROS1 non-small cell lung cancer. And the mechanism is very simple, that these drugs just interfere with the activity of the ROS1 receptor. And instead of being hyperactivated as they are in the rearranged situation, they become suppressed. And in my patients that have ROS1 abnormalities, the majority of them are thoracic patients. It is truly remarkable compared to giving traditional chemotherapy. You give crizotinib or entrectinib, and their tumor just melts away and remains smaller for such a long period of time. On the right, in the interest of time, I won't go through in too much detail, but this is also further work that is being done to say that in patients that receive crizotinib or entrectinib, because they have an ROS1 fusion or rearrangement, there are different point mutations that have been identified. So not only do we know the mechanism by which a drug will be effective in a carcinoma of unknown primary or a neuroendocrine carcinoma that potentially may be of thoracic origin, we can also have a good prediction of why something will eventually stop working. And this is helpful for those of us in drug development, that we can say, is there a new ROS1 targeting agent that can overcome the fusion, the point mutations that enhance resistance to crizotinib or entrectinib. So this is the information that we give back to the treating medical oncologist, and I'll leave it back to Dr. Al-Ghayani to continue the conversation.
Thank you very much, Dr. Khan. So actually this patient, when we got the result of the molecular tumor board, she has ECOG 3. She was in severe pain, and actually I was planning to discuss with the family the goal of care before we got the result. However, she was started in crizotinib once we got the result, because the medication is available in our hospital. And she has really remarkable response for almost 3 years, with almost complete response of her disease. Then early this year, she developed some area of mild progression. I planned to treat her with some radiation to this area, but the patient was anxious and she went to other line because she's a pharmacist and she's aware about the drugs that works in this type. So she was started on lorlatinib and she's still on therapy, and actually she just got married 2 weeks ago. So this is the end of the case, and I think this is the end of our molecular tumor board, and thank you very much.
Thank you, Dr. Mohammed Al-Garni. I'm really enjoying this evening or afternoon session. I learned a lot. I'm pretty sure that our attendees, they have the same feeling as well. So we finished 2 segments so far, and we will go for the 3rd segment with Dr. Mohammed Al-Garni. He will talk about germline oncology in the next 25 minutes. Dr. Mohammed Al-Garni, he's the one of the medical oncologists at National Guard in Riyadh. He's the director of cancer screening program. Dr. Mohammed, mic is yours.
Thank you very much, Fahad. So I think this topic might be different from what we are looking for today and tomorrow. So because when we talk about precision medicine or precision therapy, our focus tends to how to treat people based on their molecular signature. However, when we really talk about personalized and comprehensive cancer care, this is our aim to provide to each individual a personalized care that started from prevention until the end of the treatment, whether the patient survived or the patient went through the end of life care and death. And the prime example of application of personalized cancer care across this continuum is the hereditary cancer syndrome. So hereditary cancer syndrome, the classical way of looking to it is knowing what is the patient risk or the individual risk and provide some screening recommendation. However, with so many years of understanding the value of information that we get from germline genetic tests, now we use this information for cancer risk assessment. We use the information of genetic testing for prognostication and as a predictive for certain therapy. Also, based on each mutation, we provide personalized intervention and screening. And most importantly for patient, it has therapeutic intervention. This is the classical example of BRCA gene since the discovery over 3 decades now. We move from using the BRCA gene to predict cancer risk and provide cancer risk reduction strategies till we use it in the metastatic setting as a part of the treatment for those carrier of this germline mutation. And things move further, and now the PARP inhibitor are being used in the adjuvant setting for early breast cancer. So as you can see, this is one example how you can implement the use of germline testing across cancer care continuum. Always clinical case give also more insight about this. So this is one of our patients. She's a 32-year-old woman, nurse, referred to our genetic clinic with a diagnosis of locally advanced breast cancer, infiltrative ductal carcinoma, ER positive and PR positive, HER2 negative. She's a mother of 2 and she's done with her family planning. She gave a history of ovarian cancer in her mother and she died from the ovarian cancer. And when we saw the patient in the clinic, she was started already on AC protocol, and we ordered for her because the patient meets the testing criteria for germline testing. And her genetic testing showed that she is a carrier of pathogenic mutation in BRCA2 and ATM gene. And by the time we got the result, she already finished 3 cycles of her chemotherapy EC. And unfortunately, there was no clear significant clinical or radiological response to chemotherapy. So how we would use this information that we get for this patient to guide her therapy. So based on the information that we got, so now we know that the homologous recombination pathway, which is she has 2 genes and she is carrying a 2 pathogenic mutation in one of the genes included in the homologous recombination pathway, it has a predictive role to the response to platinum-based chemotherapy. So after we discuss with her treating oncologist, we plan to switch her therapy to carboplatin and Taxol. and she responded nicely to the chemotherapy. And in terms of therapeutic implication as well for the patient, given that she had 2 mutations that increase her risk of breast cancer, she already developed breast cancer, she has high risk of developing secondary contralateral breast cancer, we decided to, instead of doing mastectomy, is to go with bilateral mastectomy at the time of surgical intervention. Furthermore, given her family history of ovarian cancer and her mother in her mid-30s, we decided to go with a bilateral salpingioophorectomy, not only for ovarian cancer risk reduction, but also to reduce her cancer of breast cancer. So this is just one example how the information that we can use can change the patient outcome. And in the clinic, we always look to things that we rely on or we use to decide which patient should be tested. We used to use the risk models. There are testing criteria guidelines. There is sometimes based on pathology. And we think that hereditary component represent 5 to 10%. But I think with the data that emerged, especially over the last 3 years, we think that we are missing a lot of patients by sticking to the classical way of doing genetic tests through the guidelines. I'm going to show you some of the evidence about how the guidelines might limit your ability to detect patients with germline mutation. So this is a study that looked to more than 2,900 patients, and they did germline testing for them based on without looking to the guidelines, actually, or sticking to the guidelines, and found that if the magnitude of benefit or incremental increase to detect more germline mutation increased to 6.4%. And among those patients who were detected to have a pathogenic mutation, 1/3 of them, they have modification of their treatment based on these results. Another study that looked for cancer that they don't have screening guidelines. So now we have screening guideline for breast, ovarian, prostate, pancreatic, but there are certain type of cancer that they don't have a screening testing guideline. For example, GU cancer, for example, lung cancer. So this is a data for more than 3,000 patients. And they found on those patients, 7.3, they are carrying a pathogenic mutation or likely pathogenic mutation. And if you look to the mutation, you find that BRCA2, ATM, these mutation that has therapeutic implication for patients. So this is even for patients who with cancer that usually we don't have a screening guideline, or we think sometimes they don't have hereditary components. Another 2 studies that published this year for patients with esophageal cancer. We don't think usually the esophageal cancer commonly associated with hereditary cancer syndrome. However, these 2 publications confirm that even for patients with esophageal cancer, there is association with germline mutation, and the commonest mutation, interestingly, is the ATM mutation, which is a part of the HRD pathway, and this would explain the impressive response that you see sometime to platinum agent when you treat patients with esophageal cancer. Now, 4 weeks ago in the ASCO plenary session, they present the data about patients with lung cancer. In my clinic, actually, before this, I don't use to consider lung cancer as one of the association with hereditary cancer syndrome. However, interestingly, in this study, they screened more than 7,000 patients, and they found that 14.9% of those individuals, they carry a pathogenic mutation. And more interestingly, about 2/3 of those patients, they have a pathogenic mutation that has therapeutic implications. So this gives you more and more data that the germline testing should be part of our assessment for our cancer patient. This article published yesterday by the ASCO, and they looked to, and actually they are recommending to have a universal germline testing for all patients with solid tumor. Now, it's as a recommendation, it can be something easy to make, but the application of this recommendation is the difficulty. And all of this, since we know now the importance and the indication and these things, brings us to a very important point, which is how to improve the access to genetic testing. Now, this is the current testing, at least in my practice, what I do, universal testing. I do it for all colorectal cancer patients, for Lynch syndrome, all triple negative breast cancer, all pancreatic, prostate, and high-grade serous epithelial ovarian cancer. We do the testing for those patients. So from what I have just presented, there is increasing indication for genetic testing. Numbers of genes that need to be tested is increasing. The universal germline recommendation is growing. The therapeutic implication is increasing. And since we are doing comprehensive genomic profile, we might need to do also more of genetic testing. So how we can match the growing indication for genetic testing with and improve the access to genetic testing. Now, even in the place where there is a very good healthcare system or access in terms of number of genetic counselor and genetic clinic and genetic counseling program, also there is a minority of patients of cancer, they undergoing genetic testing. These are the data from the United States. So if you can look for a patient with ovarian cancer, which is known to be associated with hereditary cancer syndrome, only 10% of patients undergoing genetic testing. And this is also study looked in 10,000 medical record in the United States patient in COBE accredited. So it's a quality assurance project. And they found that only half of patients meet testing criteria for breast cancer, for genetic testing with breast cancer have been tested. And the situation is worse for patients with colorectal cancer. So the first step that we approach a patient when they refer to our clinic is doing the genetic counseling. So I think how to improve the genetic counseling is the key to facilitate for the access to genetic testing. But as you can see here, I don't think a busy medical oncologist in the clinic will be able to go through all this information and collect the information in his clinic about this. And this brings us to more of need of doing something that can increase the pressure on the oncologist and at the same time facilitate the test in a timely fashion. The problem with genetic testing is not like the comprehensive genomic profiling. When you do the comprehensive genomic profiling, you are looking for a specific patient for a specific mutation. And if it's there, you treat your patient, has no consequences. The genetic testing or germline testing has consequences because if you test the patient and you find they have a germline mutation, this means that you need to do family screening and testing and recommendation, which is a huge task. So there are different ways of looking to this. One of them is the classical way, which I think most of centers are still doing this by referring the patient to a specialized genetic service. This is applicable in most places in the kingdom and outside the kingdom. The problem with this system, when you refer the patient, of course you will get specialized consultation, but that the waiting time has impact. You imagine the patient that I presented with breast cancer would wait for 2 years to be seen in the genetic clinic. The other way is to do the clinical cancer genetics at every oncology unit. It's something feasible and can be done. And I think this is one of the example that can work better in our country. The other thing is to involve the oncologists in what they call mainstreaming of genetic testing. What the mainstreaming mean that each treating oncologist, for example, someone treating gynae patient, the oncologist will initiate the test in the first visit, and the disclosure of the result will be done by the genetist or by the genetic counseling. So this will save the patient the time to be seen by them, and by the time they will be seen by the genetic counselor, they will already have the results available. Other available and alternative models is the telegenetics, where you can do pre-test telephone counseling, and you can do the results, virtual clinic with the patient, and you can sometimes do a mix of all of this. And this is what we do in our center. We have a mix of all of this because the number of patients are increasing. So what we can do, we provide a genetic counselor as a physical visit first time. However, for the follow-up and for the disclosing the result, we have a virtual clinic for the patient, which actually make it easy for the patient and for us. The telegenetics has been there for some time. It was studied in different studies, and they found it's non-inferior to the physical or having the patient physically available for genetic counseling. However, the only downside of this, they found that the patient has the phone consultation or follow-up. They are less likely to do their risk reduction intervention, for example, compliance with mammogram and other screening recommendation. The growing models that we can see in the, at least in Canada, this is a Canadian study, they looked to the oncologist-led model where the oncologist is the main or the key player in the system, where they can do genetic testing based on their treating disease site, and they will arrange a follow-up visit with the genetic counselor after they get the results. Again, this is to show you that it's comparable to the physical consultation, but at the same time, the patient feel more comfortable when they are doing the genetic testing in a timely fashion rather than waiting. This is an Malaysian study. I presented it because the access to genetics testing is almost similar in the Asian population. And they found in this study where they allow the gynae oncologist to order the BRCA testing in a study, they call it MAGIC. They found the mainstreaming was effective. Of course, it has advantage and disadvantage. Mainstreaming improve the access for sure, reduce the distress and cancer worry, because the one who ordered the test is the one who's treating the cancer, so the patient have more access to information. And when they looked to the referral to the genetic specialist, for sure it will help them more in the decision making, but it will increase the risk about the cancer worry, and definitely it's poorer access for the individual. So one of the thing that I think we should try to do it in our centers is try to involve the oncologist more in the testing process based on whatever the available resources and capacity. So for sure, there is a challenges that I already mentioned in my talk. However, the possible solution is, as I mentioned, the mainstreaming or increase the use of telegenetics. So my take-home message for this is to understand that the germline oncology is evolving across the cancer care continuum, and there is a growing indication for the universal germline testing that is not only related to cancer risk assessment, but also for therapeutic implication. And we need to have more of innovative approach in order to meet the demands that we see in this field. And by this, I conclude, and thank you very much.
Thank you, Dr. Mohammed, for this beautiful presentation about germline oncology. I think we'll stay with you because I think you will run the satellite symposium sponsored by Roche. So back to you, Mohammed. We will have the questions and discussion after the satellite.
So there is a poll. There will be a poll. You want to do it now? So I would like to welcome you to the satellite symposium sponsored by Roche. This will be about the comprehensive genomic profiling in patients with colorectal cancer. We have two speakers that they will try to convince us whether to do it or not to do it. We have Dr. Canaan, she's a medical oncologist, and he's a GI medical oncologist. And we have also Dr. Mirfat Mahrous, a consultant medical oncologist at Prince Sultan Military Medical City. So as I understand, there is a poll. Okay. To whom? Okay, so there will be a link that will be sent to all the attendees, and please click on the link and answer the poll. You will find it in the chat box. Look at results. So the first question was, would you consider next generation sequencing prior to systemic therapy as baseline in colorectal cancer cases? So 46% they said yes to all my patients. 50 still running, huh? Yeah, so I think 2/3 they said yes, and 1/3 said no. So I think Dr. Mirfat, you have to do more job, presentation to convince them. Okay, so we'll start with Canaan. Okay, Dr. Mirfat, please. Okay, ladies first.
So thank you so much, Dr. Shelgarni, for introducing me. And I would like to thank Dr. Kanaan and Dr. Ashwar and all the team of National Guard for inviting me, and congratulations for this high bar scientific events. And it's always my pleasure to be with you. I would like to thank Roche for sponsoring this meeting and very exciting debate. Actually, I will make my job, it will defending the evidence, and it will be against use of CGP, comprehensive genomic profile in colorectal cancer. So this is my disclaimer. And of course, we know that for the colorectal cancer, we are treating them based on the stage, morphology, and the evidence for this. And when we look at this bar, you found that those patients, they are increasing year after year. However, when we look at the stage 1 disease, you can find that this is the best, and we have about 91% of this patient 5-year survival comparable to stage 3 and 4. And this is down even for a stage 4 disease to 12.5%. This means that we need to do a lot of job regarding these patients. But for most of the group of patients, we can see that we have a very good 5-year survival. When we look at the incidence, we can see that it's increasing, as I pointed out. I think you did not update my slides, but it's fine. Okay, because I think it's different. This is the old one. So when we look here, we can because we can see here that this is the NCA guidelines regarding what we have. So for treating colorectal cancer, we have the RAS mutation, and we have the PRF and the MSI. And this is the evidence how to treat our patients. And this is how we are starting giving them the treatment. So for any patient with the colorectal, we are doing for them the RAS. And when we have even the RAS since 2011, we down stratified the RAS to KRAS and NRAS, and followed by the PRF. This is why, because when you are giving the patient treatment anti-EGFR for patient with RAS wild, if this patient with the PRF muted patient, they have no benefit from the treatment. So the bottom line that all that we are doing regarding that the genomic part or the biomarkers, it's connected about what we are giving regarding the treatment, or even as a predictive factor. And even when you look here on the down part, you will find that the HER2. So the HER2 for a patient with RAS wild, we cannot give them also anti-EGFR because this patient has no benefit from the treatment. So this will give us the element that any biomarkers we are doing for the colorectal patients, it's based on what we are giving them as a treatment, cost benefits, and as an expectations for this patient as a sequence of treatments. And I think this is the main issue for regarding that the biomarkers for the patients. Of course, we had fantastic lectures about the CGP and the germline mutations and how we are doing the genomic profile for all the patients. We cannot ignore this part, and I think Dr. Karan will defend it very nicely. But my point that when we are treating the patient now, we have to have a validated test. We have to have a vision when you are having any biomarkers for the treatment or as prospective parts. One of the, even the MSI, it was a debate 3 years ago about doing the MSI for all the patients. But once we have launched the keynote 177 for this patient, now for all the patients with metastatic colorectal cancer, even if it's a small population, we are doing the MSI for this patient as an upfront because it's a detrimental for the treatment line for this patient as an upfront or what to do for them. So when we look even for the ISMO guidelines, we can see that even it's very old, they didn't update it since 2016. You can see that they stratified the patients between fit or unfit patients, and for the patient regarding RAS mutant, wild, or a patient with PRAF. And if you remember that we had this debate 4 years ago regarding PRAF, patient do it or not to do it, and most of the people, they are voting not to do it because there's no benefits. But once we have the TRIB study, and also those patients, they have a very bad prognosis, so it was detrimental from the start that doing PRAF, it can be give the patient the triplet therapy versus doublet, or this patient cannot be given anti-EGFR. So bottom line, when you are doing biomarker for the patient, you have to have a vision what to do for them, because if you are doing a complete panel for this patient, it will be cost. And the second element, that what to do for the other element, you don't have a treatment for this. And when we have here, we can see that based on what we have chemotherapy, and we have also the anti-EGFR treatment. Again, this is how so we started first with the KRAS, and then with the PRIME study, we have the NRAS, and even for the patient with NRAS mutant, we cannot give them anti-EGFR. So for the NTARC and the HER2, still we have a debate regarding this patient, even as a small population, but it's recommended for the patient not as a first line or as an upfront. It can be also as a subsequent line, or a patient with MSI, they have a benefit that you are maybe and needed. When we look at the MSI, we found that the MSI for early stages, it's about 15% comparable to 4%. And even MSI for the patient with early stage can be determining because they have a good prognosis, and this patient can be omit for the chemotherapy for them, specifically for a stage 2 disease, low risk. You cannot, no need for them to give them chemotherapy. And then the main part for the 4% of the patient's MSI for the metastatic colorectal, we need to give clear benefit for the patient with MSI. So bottom line, biomarkers for the patients, it needs to be first validated, have an effect, and we have data for these patients to give treatments. And for the rest mutated the patient, we know that this patient, they may have a resistance, and so they are not candidate for anti-EGFR. PRAF patient, as we are pointed out, they have a very bad prognosis. Most of them, it's about 4% of the population of metastatic colorectal. They have mostly they are from young age, and they are on the right side. When we look at the HER2, as I pointed out, for a patient with RAS wild, so for patient with RAS mutant, we are not for them the HER2. For RAS wild patient, you can do for them HER2 amplification, and we have established a protocol for this patient, and you can give doublet. So the HER2, you can give them a doublet HER2 blockade, trastuzumab, pertuzumab, and it's validated and based on the Hercules study. Again, the right side and left side, we know that they are different as a prognosis, and those patients in favor to give them anti-angiogenic comparable to anti-EGFR. And we know that patient with right-sided, that they have a bad prognosis comparable to left-sided. And I will not spend so much regarding the ctDNA. It will be covered correctly by nicely by Dr. Karan, but we have to know that the ctDNA, it's one of the coming, but still not validated to be used. And we are looking much for this. Before I'm going for the last slide, that we have to know that we are talking about the evidence, we are talking about the cost, And what we are doing, if we do have a panel of biomarker for the patient, and so the established for us based on the NC guidelines, ISMO guidelines, and ASCO is RAS, HER2, and NTARC, and of course the PRF. And this is what we are doing regarding patient and what we have validated test, and of course MSI. Thank you very much for your attention.
Don't jump to conclusions, Dr. Fahad. Well, first of all, I'd like to thank Roche for funding the satellite symposia, and I'd like to thank Dr. Mirvat for making this very exciting and making it difficult for me. So I will debate pro or for performing next generation sequencing in metastatic colorectal cancer. This is not working. So I have divided up my talk, and I really would like to hear the opinion of my GI oncology colleagues in the end into the these three following subtopics. So I'll talk about MSI and tumor mutational burden. Then I will present probably a debatable approach on how to approach microsatellite stable metastatic colorectal cancer. I'll end by talking about interact fusions. So TMB data and overlap with deficient MMR. Now I know Dr. Riham spoke about this in her talk earlier, but microsatellite unstable tumors are not equal. Now immunohistochemically, we test for MSH2, MSH6, MLH1, and PMS2. And we know from this study by Hall, which was presented in ASCO GI, that loss of MSH2 and MSH6 may be associated with a higher TMB score, as opposed to loss of MLH1 and PMS2. Now, this is when this is tested using immunohistochemical IHC. Now, when we look at the tumor mutational burden across all GI cancers, this was published by Dr. Salem in 2018. So it's actually older data. We see the right-sided colon cancer having had almost 12% with TMB scores of more than 17%. This is quite exciting because we do have a pembrolizumab being approved for TMB more than 10 in pan-cancer or pan tumors. So MSI high tumors most likely are TMB high, but not all TMB high tumors are MSI high. So are we missing certain patients who may have tumor mutational burden of more than 10 if we don't test for TMB specifically? Now, this is from one of the keynote studies, and what I want to show here is that they have tested quite a large number of CRC patients. Now, patients who were TMB high, so TMB of more than 10 mutations per megabase, but were not MSI high, were actually 18. So they missed 18 patients if we only treat patients with pembrolizumab or immune checkpoint inhibitors, if we only treat them if they were MSI high. So this does provide some sort of evidence that we should be testing for tumor mutational burden. Now, how do we test for TMB? It is currently only assessed by a large panel of next generation sequencing. Now, TMB varies significantly across MSI high tumors. I have spoken about that the loss of expression of MSH2 and MSH6 may be associated with a higher TMB score, as opposed to loss of MLH1 and PMS2. Now, when we look at the average TMB score in all colorectal cancer cases, now this does not really divide it up into left versus right side, the average is just a little bit less than 10. However, I have shown you that right-sided tumors perhaps have higher TMB score. Now, pembrolizumab has been assessed in many trials. The 158 one basically gave it its approval, its FDA approval for TMB high, more than 10 mutations per megabase in all solid tumors. And it is also FDA approved in microsatellite unstable tumors. Now, this is, I think, where we're going to have a bit of a debate, how to approach microsatellite stable metastatic colorectal cancer. So if we divide them up into RAS wild type and RAS mutant and sidedness. So if a patient has, now this is metastatic colorectal cancer, if a patient has RAS and BRAF wild type and is left-sided, I think we have very convincing evidence that we should be giving them chemotherapy, now whether doublet or single agent, and an anti-EGFR monoclonal antibody. If the patient is RAS or BRAF mutant, or if the patient is right-sided and is wild type, we perhaps should be giving them Avastin rather than an EGFR monoclonal antibody. However, we know that there are mutations that may confer resistance to anti-EGFR monoclonal antibody. There have been quite a few publications about this. HER2 amplification retrospectively is one of them. MET amplification is also one of them. EGFR extracellular domain mutations are one of them, and some others. So this portion of mutations are known and have some level of evidence that may actually provide resistance to anti-EGFR monoclonal antibodies in the first-line setting. Now, a very interesting study done by Chiara Cremolini, who's a physician scientist in Italy who specializes in colorectal cancers. So she basically created a limited gene panel, NGS panel, and she chose basically genes that are associated with resistance to anti-EGFR monoclonal antibodies. She took patients who were given anti-EGFR monoclonal antibodies. Now remember, this is 2017, so this is before the sidedness data. She found that some patients were sensitive to anti-EGFR monoclonal antibodies. Some patients were resistant to anti-EGFR monoclonal antibodies. And she did this panel, which she called a pressing panel. So this limited panel NGS. So she found that there were HER2 amplifications or mutations, MET amplifications. Now, in also colorectal cancer, there were ALK, ROS, NTRK, and RET fusions, and PIK3CA, PTEN, AKT, and RAS mutations, which were found at low variant allele frequency of detection. And all of these mutations were associated with resistance to anti-EGFR monoclonal antibodies. So she looked at resistant patients and sensitive patients, and 20 out of 47 patients, so probably 40%, had basically positive pressing panels. So they had one of these mutations, which conferred resistance to either cetuximab or panitumumab in the first-line setting. Now, you may say that maybe this difference is according to sidedness. Maybe all of the resistant patients were actually right-sided. However, when you look at this assessment here, when you look at this black rectangle here, these are left-sided patients who have had detection, like this one had a HER2 mutation. This one had a PIK3CA mutation and is left-sided tumor. This one is a left-sided tumor with low frequency of RAS. These were all resistant patients, with a significant number of them being left-sided rather than all of them being right-sided. You may say that, okay, if we don't give EGFR monoclonal antibodies in the first line, we're going to give it in the second line, and overall survival is going to equate. But that's not the case. So another study done by Murano, I think also in Italy, this was an assessment of Valentino, which looked at panitumumab in RAS and BRAF wild type. They looked retrospectively at pressing negative and pressing positive patients. So NGS pressing positive, and they found that the progression-free survival was actually significantly less compared to pressing negative NGS. Overall survival was also significantly less if you have pressing positive, regardless of sidedness. Let's look at the sidedness topic more carefully. So this was the initial pressing NGS study. Left-sided patients actually equated to 20% having this NGS positive to a resistant mutation that is predictive of non-response to EGFR monoclonal antibody. Also, the post-hoc analysis of Valentino also found 1 out of 5 patients having pressing positive NGS. Let's look at the post-hoc survival analysis of Valentino and look at the sidedness data. So if you are left-sided and this NGS is negative, the progression-free survival is 13 months. And if it's positive, it is significantly less. It's around 8 months. If you're left-sided, which we assume that they are the ones that will respond to EGFR monoclonal antibodies, regardless of whether we do NGS or without. If they have a pressing or a limited panel NGS is positive, their survival is actually less by probably 20 months, which is quite significant. So, and I think this is the debate, and I would be interested in hearing your opinion, Dr. Fahad, Dr. Mohammed, and Dr. Shawqi, and you, Dr. Amirrat, all the GI oncologists, is should the approach be like this for our first line, microsatellite stable, metastatic colorectal cancer, so RAS, BRAF wild type and left-sided, and this limited panel NGS pressing should be negative, because if it is positive, I've shown you some evidence that progression-free survival and overall survival is actually less compared to pressing negative, regardless of sidedness. Now, the recommendations from ESMO do mention that maybe we should not be using NGS. However, and this is the 2020, so it's a little bit old, it does say multi-gene NGS can be an alternative to PCR tests only if it does not generate extra cost. Now, let's talk about the NTRK data. So we have 2 drugs, larotrectinib and entrectinib, and they are approved for solid tumors with an NTRK fusion. Now, NTRK fusions are quite rare. We know this. From the entrectinib data, 10 patients out of the study that was published in 2020 were actually colorectal cancer patients. Now, when we look at the larotrectinib data, colon cancer, so the pink columns, impressive waterfall plot, most of them responded. So there were patients with colon cancer that benefited from larotrectinib. I want to skip the slide, but clearly you can see clear benefit in terms of overall survival, progression-free survival, and duration of response when for NTRK fusion positive tumors. Entrectinib also included patients with colorectal cancer, and these patients also responded based on this waterfall plot. So how do we test for NTRK? And there is the IHC test, which gives you rapid results. It is relatively low cost. It may deplete tissue, and it is less well validated currently. There is the NGS, which has its advantages by testing more genes, specifically the pressing panel, which I have explained that may confer resistance to anti-EGFR monoclonal antibody. It is still perhaps costly. However, the fusion partner position is well defined. So it is a more validated and a more solid test to detect NTRK fusions. Who should we test for NTRK fusions in colorectal cancer? I've taken 3 publications. All of them are probably a year or a couple of years old. So NTRK fusions are present in less than 1% in all colorectal cancer patients. Some studies, in fact, report 0.3%. So 3 out of 1,000 patients being tested are going to be positive. So they are quite infrequent. However, when this test is enriched in your microsatellite unstable patients, it can be up to 7%. And I've seen reports, in fact, reporting a higher number than this. So we probably should be testing all our microsatellite unstable patients for NTRK fusions. And I have shown you that NGS has a more validated and a more solid test compared to IHC. In conclusion, should we do NGS testing for all of our metastatic colorectal cancer cases? I have put yes, but I have added a statement. I said, if the technology is available and does not incur significantly increased costs. Now, remember, we are in 2022. Vendors are increasing who can actually perform NGS. I know hospitals, certain hospitals have the technology in-house as well. We should strongly consider developing in-house technology, and we should consider, and I put consider, doing pressing limited NGS upfront and consider anti-VEGF inhibition for pressing positive patients rather than giving them an anti-EGFR monoclonal antibody upfront. Now, I think we all know the story with pembrolizumab in tumor mutational burden high. And I have shown you that TMB high colorectal cancer data actually exists. So we should be testing TMB in our colorectal cancer cases. NTRK fusions should be tested in microsatellite high metastatic colorectal cancer. Responses are remarkable. So we should be testing these patients for NTRK fusions using NGS. I do think that the cost of doing KRAS, NRAS, BRAF, HER2, MSI, and perhaps others, and I know that this list of biomarkers will increase with time, is perhaps going to be more costly if we take each test alone compared to an NGS panel. And I can see a pathologist in front of me nodding here.
I will give you a comment at the end.
Oh, okay.
I will give you a few.
Right. Thank you very much. I think we should start the discussion. Yeah, for all, should we? Yeah. I will say.
I would like to thank all our speakers for these beautiful presentations and overview of a topic that's increasingly interested. And with time, we get more insight and we're learning more and more. Probably it's the way to go in the future. Still, we had some debates throughout the discussions that we all listened to. So we'll start some discussions. We still have maybe 10-15 minutes, so we try to utilize our time very well. So maybe I'll start.
I have a question, Dr. Fahad.
Go ahead, Dr. Shawqi, تفضل.
Okay, so I have a quick question to Dr. Kanaan and question to Dr. Muhammad. The question to Dr. Kanaan, I thought the number of patients, first of all, he didn't give us what the number of patients. All what we have seen is percentages. So how many patients they have done, we really don't know. I would rather if he can tell us the number of the patients. But the question is, he mentioned that 90 plus of his patient had targetable, of the patients that were discussed had targetable mutation, or maybe I misunderstood, and maybe 80 plus have the medication that can be targeted. I thought that's a little bit too high. So are they picking for discussion those patients who actually have positive results and just those with completely negative NGS, they are just discarding them? That's the first question, Dr. Khan. The second question, Dr. Muhammad, is so patients who have germline mutation like BRCA mutations or like the Lynch syndrome, those patients are susceptible to more than one cancer. So it's maybe breast, colon, GYN, pancreas, prostate, etc. Who should be following those patients and assigning them for the appropriate follow-up recommendation? Because apparently each one of them, there should be a recommendation. So if somebody is BRCA positive, he has risk of prostate, pancreas, ovarian, breast, and now there are 3 more, which is biliary, gastric, and GE junction. Who is the person who should be in charge of those patients? That's my questions.
Thank you, Dr. Shawqi. So are you able to hear me?
Yes, yes, I can hear you very well. Thank you.
Excellent. So thank you for your question. So the first question is, what is the sample size? I did not put it on purpose because the data is prepared to be presented, inshallah, in a, I hope, in an international meeting. So watch the space and you will know the numbers, inshallah, very soon. Regarding the percentage that have had recommended therapies, the 90%, Dr. Shawqi, was genomic and non-genomic recommendations. The patients who got recommended genomic therapies, so based on their genomic alterations, are actually 73% of all patients presented. Now, have we excluded NGS negative patients? Yes, we have. Who have we included? I think I've made it clear in my presentation. Patients with complex genomic profiles, so patients with non-tier 1 and more than 1 mutation. So these were the ones who, and in the eye of the treating oncologist, we were not able to come up with a recommendation without the help of the molecular tumor board. So I hope that answers your 2 questions. Okay. And I'll give the mic to Dr. Mohammed now to answer your question.
Thank you very much, Dr. Shawqi. This is really a very important question. With all this expansion in the indication, and the expectation is to find more and more individuals carry this mutation. I totally agree with you. There must be someone who is in charge and overlook and coordinate the care for those patients. So I think it depends on an institution based, places. For example, where I was trained, usually the cancer genetic clinic, they see the patient, they make a clear recommendation about what exact surveillance should be done. And because of the effective primary care system there, they can be carried out by the primary care team. And we see them once a year just to make sure that this recommendation has been done. The situation here is different. So we don't have really a very effective primary healthcare system at national wise, but at least in my institution, we adapt the practice. And this is the idea of establishing the cancer control division, is to have multiple clinics under this division looking for those high-risk patients. We have a nurse specialist, we have a coordinator, and we have a multidisciplinary team that we coordinate with other services. So we need to coordinate with the scope endoscopy unit. We need to coordinate with the gynae oncology for prophylactic surgery and these things. So for the meantime, I think it depends on the institution and the available resources. But what we adapted in our center is to have a dedicated program runs and by myself, where we can have a dedicated clinic for those patients. And we don't have really high number of, because we don't keep all the patients. What we do is once we find someone carry a pathogenic mutation, if it's a high penetrant, which means that they have a very high risk of developing cancer, those patients, which I take care of them, and those patients that we do family screening. But if it's intermediate or low risk gene, we provided them with the medical reports, with the screening recommendation, and they can follow where they are eligible. But I agree with you totally. This is unmet need for our population.
Thank you.
So I have another question again to Mohammed and Kanaan about the molecular tumor board. I think the whole debate about the use of molecular tumor board is that these recommendations, they're not coming from, outside the tier 1. It's not like solid high-level evidence. That's the source of all the debate. So from your experience in your institution, tier 1 will goes to like lung tumor board, GI tumor board, of course, but then controversial recommendation goes to molecular tumor board. How comfortable as clinicians that you gather and recommend some treatment that maybe some will consider kind of experimental, and then how would the let's say the hospital itself, the institution, will provide this treatment based on this recommendation, because obviously it's not stated in any guidelines, most of guidelines.
Thank you, Dr. Fahad. Yani very important question indeed. So how do we, I think your question is how do we ethically give patients these questions, how do we recommend these therapies? So if you recall, I have mentioned that these patients are chemorefractory patients, Dr. Fahad. So these are patients who, without therapy, we know that their survival is going to be perhaps around 3 to 6 months. This is historical data and very well-known data. So performing NGS and having these sort of non-tier 1 recommendations does provide them with some sort of therapy line. I know that it's not really based on high-level randomized controlled trials, and it's not really based on high-level sort of evidence. However, I think I have shown you and shown the audience that it does work. And we have had, chemorefractory patients who have had median progression-free survivals of 9 months. Case that Dr. Mohammed ROS1 alteration in a neuroendocrine tumor. We would not have found this mutation unless actually we performed the NGS and discussed it in a tumor board. The case that Dr. Samah has treated in National Guard Jeddah, and pancreatic pseudopapillary tumor, receiving everolimus and tamoxifen, and remaining on therapy for almost 1 year. So I agree with you, it is difficult. However, and let's remember that we are providing an incremental benefit in a population of patients that are in great need of any sort of benefit. Not sure if Mohammed, you have any additions.
So I agree with you, Fahad, because when you request this medication, which is really expensive or not included in the guidelines, you find always challenging in getting this medication or approval from the. But what we are trying to do, actually, this is a part of the precision medicine, the new department established in the National Guard, which is genetic and precision medicine. And one of the things that we did also, we have an umbrella protocol for precision oncology. And it's an ethically approved by the institution where we can do the genetic testing and use the most appropriate therapy based on this recommendation. In addition to that, we have a consensus with the pharmacy in terms of this is a conclusion of molecular tumor board. They will provide the medication.
دكتورة ميرفت.
عفوا. اتفضل يعني.
I think he needs the mic.
Thank you very much. Quite excellent presentation, excellent debate. So just 2 points. 1 point regarding the testing. If you factor in the cost for the MSI, you need 4 markers. Each one of them costs 400 to 500 reals. You're consuming the material, pathologist, technologist time, and add to that the test for each one of them. Compare that cost to Foundation 1 and see how much, or an NGS panel, let's say, like maybe 60 or 100 gene panel. So that's one factor. The other factor is that there is recent approvals for liquid biopsy with Garden 360 and Foundation 1. So what are you going to do with the liquid biopsy NGS data? You will have to go back and test the tumor on the tissue to compare. So in the end, I'm not being zealous when I say that maybe at a point of time, one of the requirements of establishing good oncology service is to have an active and running NGS service. Maybe they will get a better technology in the future, maybe soon future, but that's, I think it's becoming more standard. For NGS, if you review the guidelines also recently, they recommend NGS versus other methodologies for PCR. So these are my few notes. It will becoming less and less quality. And the problem is now dealing with the tremendous amount of data with the bioinformatics pipeline, but they will solve that in that way. We can still do MSI with NGS now. They have modification with their software in which they can do MSI, they can do human mutation burden, and the huge number of things. So originally I'm a proponent of selective testing and knowing the answer before the question. But in this one in particular, I think that NGS is already very important. And maybe at some point.
So how do you explain that in ESMO, they are not recommending, and also in the other guidelines, they are still not recommending NGS as an upfront and still recommending only the validated biomarkers? You pointed a very important point regarding the cost, because it's really NGS will give me the whole information, but I'm testing the specified one because it's detrimental about the treatment. But when you are pointing now, the cost NGS comparable to what we are doing, this means less. Yes. Okay.
But also the AstroCap guideline, most recent one for lung.
Yeah, lung is different entity because we are in lung, we are doing the NGS. This is established simply because every gene we do have a treatment for this. But in the colon, yeah, and strong evidence. But in the colon, we still not. Yes, we definitely it's give us an understanding of the biology and biomarkers. And as Dr. Kanayan pointed out, there is even can give you a hint about what to give regarding the treatment as an upfront, even if we do have every. But in the lung, it's a different story.
My second point is that with the molecular tumor board, I think that's also one of the prerogatives of the molecular tumor board, is to indicate which technology or the standard of treatment available for the history. So it has to be agreed upon.
Yes.
What's the platform? What sort of data we're going to handle? And what to expect? So
You're absolutely right, because we are an area in between. This is considered that understanding and the biological parts we needed to have a figure about it. And the other part, this is an information how I can use as a treatment for our patients. And we are sitting in the area in between, and we are stick to the evidence and the validated test. This is why we are area in between. And this will bring me to my question to Dr. Kanayan. You pointed out the TMB, and the TMB, not only in the colorectal, in a different disease. We do have different tests and still not validated to be used as an entity as a predictive for the treatment of IO therapy. We know that patient with TMB more than 10, and in some studies more than 12, and even some studies more than 18, those patients, they be candidate for IO therapy. But as a separate entity, it failed to show that comparable with MSI or PD-L1 positive patients, that those can be used as a separate entity for use IO. We do have for the colorectal, the Keynote 177 as a phase 3 study and established, and also we have a FDA approval. We don't have FDA approval or approval by the guidelines for a TMB as a separate entity. So what do you think?
Thank you, Dr. Mirvet. So pembrolizumab is actually FDA approved in TMB high, more than 10 mutations per megabase, pan-cancers, after failure of standard therapies. This is actually an FDA approval, and this is irrefutable. Now, I'm not sure if your question is asking specifically about metastatic colorectal cancer cases. Yeah, so in colorectal cancer cases, I know evidence isn't great in terms of the number of publications that actually speak about TMB high. However, I have shown you some evidence. There's evidence done by Dr. Salim, and it actually dates back to 2018, 2019, where right-sided colorectal cancers actually have a higher TMB scores. We know that there is overlap between MSI high and TMB. So most MSI high tumors are actually TMB high. However, not all TMB high tumors are MSI high. So the debate is, are we actually missing a small subset of patients who are TMB high, so TMB more than 10, who have an FDA approved drug, after failure of some sort of lines of therapy and are not caught by some sort of standard testing. Now, TMB is tested only during in some NGS panels. So you cannot really test for it using other sort of testing. And I think this is where the debate is. Now, and yes, تفضل. Okay. Sure. I actually have a question for my GI colleagues, which is actually want to hear everyone's input on the pressing data. I thought this was very interesting. In fact, I looked at it and I thought this is new information to me, that we actually do an NGS to determine primary resistant group of patients to anti-EGFR monoclonal antibodies. Now, anti-EGFR monoclonal antibodies give us great benefit in left-sided tumors who are RAS and BRAF wild type. So I'm not sure if Dr. Shawqi, you are with us. Actually, I'm interested in hearing your comment in the limited pressing panel. And I'll hear Dr. Fahad and Dr. Amirvat.
Yeah, thank you, Kanaan. So you presented a very interesting topic about this pressing panel. And again, that's the way I look at this topic, how much we are convinced about these data. I know not all oncologists, they want to prevent their patients from receiving anti-EGFR because of some anti-HER2 or something, expression or things. So I think it comes to how much you're convinced by this good, but not yet high-level evidence. To me, I am convinced. I think we should be smarter using these treatments, and I think it will be very helpful. Mervat.
Yeah, I concur totally what Dr. Fahd pointed out, and I think it goes with about 40% of the patient with RAS wild, they have a resistance regarding anti-EGFR, and this was explained by the Valentino study, as you pointed out. Even it's a small patient population, but at least give you a signal that the patient who are resistant to this treatment, we have an entity which is missing, because before we just only have the KRAS and the NRAS, and then we had the PRAF, and we know that this patient, they aren't responding, and then we have the HER2. Still, this small patient population is still resistant, and think this is explaining why this patient so it gives us a validation that maybe this patient needs to go for this as an upfront, and again, this gives you understanding that as a predictive factor for this patient, because we know that those patients, they are not responding to anti-EGFR, so you can expose them at the start for anti-angiogenic therapy, and you can skip giving them a treatment which is not benefits. This is why we are trying to look at those patients. What is the benefit for them to do it for as an upfront? And this is the main issue. Thank you.
One of the side effects of taking an unorthodox approach, not following the sequence that's expected, is that you might get the results that need to make you confirm. So let's say I have a scenario. Someone who attends to report, you have a patient metastatic colorectal carcinoma, and also thyroid, or a previous history of a thyroid tumor, and they have BRAF mutation, or maybe history of melanoma. How are you going to resolve this issue? How are you going to work with that? How are you going to retain your molecular tumor?
I didn't get exactly the question.
So you mean there's some mutation or things, but you don't know if it's related which primary?
Patient came, presented at your institute with metastatic colorectal cancer. You did the molecular testing and BRAF V600E mutation, otherwise nothing. And at some point clinically, or someone brought up the thought that could this be thyroid or could this be melanoma, which can have the same mutation. So you can have similar pathologies with different mutation, and sometimes different mutation with similar histomorphology. So that's a side effect of having an unorthodox approach. How are you going to avoid this happening? Or how are you as an oncologist will make a decision since you lead
I think as an oncologist, I would refer back to
molecular
my
tumor
pathologist.
Thank you, Dr. Hatem. So I agree with everything you said, except that it's unorthodox. It is becoming more orthodox nowadays. So the dabrafenib trametinib, and I actually liked that you mentioned BRAF V600E. 3 months ago, in June 22nd, there was a US FDA approval for pan-cancers. So regardless of whether it's a thyroid, whether it's a colorectal cancer, tumor agnostic indication, after at least progression on a single line, you can give them the dabrafenib and trametinib. And I think this evidence is actually progressing. One of the slides that I presented in the first lecture was on projected tumor agnostic indications. Now, do certain mutations make a certain diagnosis more likely? Perhaps, and this is where a phone call to you and your colleagues from pathology definitely helps. However, let's not forget that there are some mutations like ALK translocations, like ROS1 fusions. These mutations are very close to actually getting a pan-cancer or a tumor agnostic indication. Prime example is Dr. Muhammad's case, ROS1 in a neuroendocrine tumor, and a remarkable response for 3 years or so on anti-ROS1, basically tyrosine kinase inhibitor. So this is developing, this is changing, this is actually becoming from unorthodox to becoming orthodox.
But what about the thyroid papillary carcinoma? Is it so now we might be at a point where it's not important to know if it's papillary thyroid carcinoma, if it's melanoma versus the primary tumor that you are treating?
Yes.
No, I think still histology has value, and the prognosis of cancer, for example, BRAF mutant melanoma, it's not like BRAF mutant colorectal cancer. Even if you are using the same targeted therapy, the prognosis is different. So I think it's not only about molecular signature, still the histology and the grade of the tumor, the classical way of looking to the tumor, it still has a value. Yes.
Okay, I think we have come to the end of day one of our meeting. It was exciting and we learned a lot today. I would like to thank our esteemed speakers and moderator for their engagement and the nice discussion. I would like also to thank our audience, virtual and physical attendees. And tomorrow we'll see you, inshallah, at 9 a.m. and remind you about the parallel session for the medicine and for the hours. Thank you so much. Thank you. I will give you a minute of the topic. Very fast pace. Thank you very much.